Dear delegates, on the third day, I had a video felicitating the COVID warriors who are working day and night in the hospitals, and I failed to show it. Today, I'm taking that chance and streaming that video through my YouTube and this Google Meet. I think it is my felicitation to my students who are working day and night in this COVID situation.
In this year, 2020, in this year, 2020, the entire world changed completely in front of our very own eyes. Due to the alarming rise in cases of COVID-19 around the World Health Organization declared a public health emergency of international concern, the highest level of alarm under international law, who confirmed the coronavirus outbreak a pandemic. On 24th of March 2020, the government of India ordered a nationwide lockdown for 21 days as a preventive measure against the COVID-19 pandemic. When the entire country was under lockdown, the frontline COVID warriors were busy in their line of duty. The doctors, nurses, lab technicians, pathologists, dietitians, other medical staff, The sanitation workers, the police, and several other government officials were working day and night to serve the nation, to serve the people in this emergency situation. Diet therapy is a crucial aspect of treatment in any disease, including COVID-19. Dietitians and nutritionists have an important role to play in reducing risk of complications, shortening recovery times, and rehabilitation. Several students from the Department of Home Science, University of Calcutta, are currently working as dietitians in reputed hospitals across the country. Therefore, on the occasion of National Nutrition Month 2020, we take this opportunity to express our gratitude to all our seniors, batchmates, and juniors who are dedicatedly working as dietitians in this pandemic situation as frontline COVID warriors. We managed to contact a few of them, and we have requested them to share their story with us.
এই যে মার্চ মাস থেকে আমরা যে লকডাউনে ফেস করছি এখনো পর্যন্ত আমরা কিন্তু আমাদের একটা কোভিড এই শব্দ কিন্তু কমন আছে মানে আমরা যখনই আমরা রাতে শুতে গেলেও আমাদের এই ওয়ার্ডটা মাথায় থাকে ঘুম থেকে উঠে ঘুমের পরেও এই কোভিড ওয়ার্ডটা কিন্তু আমাদের মাথায় থাকে।
কারেন্ট প্যান্ডেমিক সিচুয়েশন আমাদের কারোর জন্যই কোনো রকম সুখদায়ক স্মৃতি নয়, তবে একজন হেলথ ওয়ার্কার হিসেবে আমার কাছে এটি একটি সেলফ লার্নিং প্রসেস। যে মুহূর্তে জানতে পেরেছিলাম আমাদের হসপিটাল কোভিড ডিক্লেয়ার্ড হসপিটাল হিসেবে ঘোষিত হয়েছে, প্রাউড ফিল করেছিলাম, কারণ সেই সময় এই অঘোষিত যুদ্ধের সৈনিক কিন্তু আমিও ছিলাম।
আমি একজন ডায়েটিশিয়ান হিসেবে এটুকু বলতে পারি যে হসপিটালে ডায়েটিশিয়ানের রোল কিন্তু অত্যন্ত গুরুত্বপূর্ণ।
যে মুহূর্তে মানুষরা গৃহবন্দী হয়েছে সেই মুহূর্তে আমাদের ছিল বাড়ি থেকে বেরিয়ে ডিউটি করার সময়।
Right now, on this pandemic situation, I am an active member of RT-PCR. So patient came for the test, COVID test, and we have to do their screening, COVID screening.
ডায়েটিশিয়ান হিসেবে প্রধান চ্যালেঞ্জ যেটা আমি পেশেন্টের ক্ষেত্রে ফেস করেছি ডিউরিং দিস কোভিড ফেজ, সেটা হচ্ছে পেশেন্টদের মেন্টাল ব্লকেজ। পেশেন্টরা কিন্তু প্রচন্ড পরিমাণে অ্যাংজাইটিতে ভুগছিল, ডিপ্রেশনে ভুগছিল, এবং সেই কারণে তাদের খাওয়া দাওয়া তারা অলমোস্ট বন্ধ করে দিচ্ছিলেন। আর পেশেন্টদের কাউন্সিলিং খুব গুরুত্বপূর্ণ ছিল থ্রু আউট দিস প্রসেস। এখনো চেষ্টা করি যে সমস্ত পেশেন্ট অ্যাংজাইটিতে ভুগছে তাদেরকে প্রথমে সঠিক কাউন্সিলিং দেওয়া এবং তাদের এই কনফিডেন্সটা আনা যে
আপনি এফেক্টেড হয়েছে, ভয় পাওয়ার দরকার নেই, যেমন ট্রিটমেন্টের জন্য মানসিক শান্তিকে বজায় রাখা এবং খাওয়া দাওয়াটা ঠিকমতো করা।
আমার কাজটা বেসিক্যালি ক্রিটিক্যাল কেয়ার ওরিয়েন্টেডই বরাবর ছিল। আইসিইউর সাথে খুবই অ্যাকাস্টম ছিলাম, কিন্তু আজকাল আমাদের ওয়ে অফ ডেলিভারিটা একটু চেঞ্জ হয়েছে, কারণ কোভিড পেশেন্টও দেখছি নিজেকে অনেক বেশি প্রিকশন নিতে হচ্ছে।
এর পাশাপাশি নানা রকম কোমরবিডিটি তো পেশেন্টদের যাদের ছিল সেই কোমরবিডিটির এফেক্টটাকে মিনিমাইজ করাও ছিল একটা অন্যতম চ্যালেঞ্জ।
ডায়েটিশিয়ান হিসেবে আমাদের ভূমিকা হলো সাধারণ জনগণকে প্ল্যানিং ডায়েটের মধ্যে কিভাবে সুস্থ রাখা যায় সেটা বোঝানো। যেরকম আমার প্ল্যানিং ডায়েট থেকে বললো যে একটা ওয়েল প্ল্যান্ড ডায়েট, একটা ওয়েল ব্যালেন্স ডায়েট খুব দরকার ইন অর্ডার টু কিপ ইয়োরসেলফ হেলদি অ্যান্ড ফিট। সেটাকে মাথায় রেখে আমরা সেইভাবেই ডায়েটারি অ্যাডভাইস দিয়ে থাকি।
ব্যালেন্স ডায়েটটাকে আমরা ফলো করবো, অ্যাবসলিউটলি হোম বেসড ব্যালেন্স ডায়েট, যেটা আমরা সবসময় রেকমেন্ড করছি। ইমিউনিটিটাকে মেনটেন করবো।
For diet, I prefer high protein diet and also immunity boosting diet. I suggest vitamins, minerals, and zinc rich food and also supplements.
আমাদের গভর্নমেন্ট অর্ডার আছে, সেই অনুযায়ী আমরা ঠিকঠাক ভাবে ডায়েট সাপ্লাই করছি হসপিটালগুলোতে। আমাদের প্রত্যেক দিনের খাবারে কিন্তু ঠিক যেরকম আমরা অর্ডার অনুযায়ী রাইসও যাচ্ছে, প্লাস হচ্ছে যাদের হ্যান্ড থাকছে কোভিড কনসেন্ট্রেশনের সেই অনুযায়ী আমরা ইউনিও সাপ্লাই করছি। প্লাস আমাদের মেন ফোকাসটা কিন্তু এক্ষেত্রে প্রোটিনটা, আমরা দুধ বেলা কিন্তু প্রোটিনটা সাপ্লাই করছি, ফার্স্ট ক্লাস প্রোটিন।
ফ্রুটস অ্যান্ড ভেজিটেবল তো ভিটামিন মিনারেলস অনেক বেশি, সেই কারণে ফ্রেশ ফ্রুটস অ্যান্ড ভেজিটেবলস পেশেন্টের ডায়েটে অবশ্যই রাখতে হবে। এছাড়া পেশেন্টের স্ন্যাকিং টাইমে পেশেন্টকে র ভেজিটেবলস, তারপরে ফ্রেশ ফ্রুটস এগুলো দিতে হবে ইনস্টেড অফ স্ন্যাক্স কন্টেনিং এক্সেস অ্যামাউন্ট অফ সল্ট অ্যান্ড সুগার।
খাদ্য তালিকায় রাখাটা দরকার সেটা হলো ভিটামিন সি বা অ্যান্টিঅক্সিডেন্ট রিচ জাতীয় ফুড,
যেমন আমলকি, পেয়ারা, যেকোনো একটা সাইট্রাস ফ্রুট যেমন কমলালেবু, মৌসুম্বি, এই জিনিসগুলো।
কারণ আমরা সবাই জানি এই সময় কিন্তু ভিটামিন সি রিকোয়ারমেন্টটা অনেকটা বেশি দরকার। কারণ ভিটামিন সি রিকোয়ারমেন্টটা দিলে আমাদের কি হবে, আমাদের শরীরে হিট পাওয়াটা কিন্তু অনেকটা বাড়বে।
এখন এই মুহূর্তে যেটা আমরা বলে থাকি যে বাইরের খাওয়া, বাইরের জাঙ্ক ফুডস লাইক প্রসেসড ফুডস বা দোজ ফুডস which are rich in preservatives, সেগুলো একটু অ্যাভয়েড করাটা এখন বেটার। কিন্তু রেস্ট্রিকশন আসলে আমরা একটা জিনিস অ্যাড করতে চাই যে প্রপার নিউট্রিশনের সাথে সাথে কিন্তু হাইড্রেশনটাও আমাদের ভুললে চলবে না। নিজেকে ওয়েল হাইড্রেটেড রাখার জন্য প্রতিদিন কিন্তু আমাদের 8 থেকে 10 গ্লাস জল আমাদের খেতে হবে।
And water plays a very important role for moving patients.
তবে হ্যাঁ, যেটা আমি বলবো যে যাই খান না কেন, যাই তাকে প্রিপেয়ার করবেন না কেন, that should be in a very hygienic form.
এই পিপি পরে যদি আমি রাউন্ডে না যেতাম, যদি এই অপরচুনিটি না পেতাম পেতাম, তাহলে আমি সত্যি বুঝতাম না যে ডক্টর নার্সরা কতটা কষ্ট করে চলেছে সারা পৃথিবীর ডক্টর নার্সরা। তারা তো 12-14 ঘন্টা এক টানা ওই পরে রয়েছে উইদাউট এসি, কোনো শ্বাস নেওয়া যায় না মাস্ক পরলে, ফেসিয়াল পরে থাকলে, মানে সত্যি ডিফিকাল্ট, সত্যি কষ্টকর।
প্রফেশনাল রেসপন্সিবিলিটির কাছে কোভিড-19 এর যে ভয়, সেটা কিন্তু সেই সময় আমাদের কাছে একটা বিশাল চ্যালেঞ্জ হয়ে উঠেছে এবং সমাজের স্বার্থে দুটির সাথে সাথে নিজেকে এবং নিজের ফ্যামিলিকে সুস্থ রাখবো, আমাদের কাছে একটা বিশাল বড় সমস্যা। সো আমি একটু আমার এক্সপিরিয়েন্স শেয়ার করবো যে those times, মানে those days initially আমরা মানে ফেস করেছি এই সিচুয়েশন, those times were really very tough and at least we are very lucky to have such a supportive family, both our hospital family ও বলছি, আমার বাড়ির family ও বলছি, যে they made us understand, they made us very well understand that yes, this is the time you should do something for the patient. For the sake of patient, you want everything to be done. So it's a positive vibe. এটা পেয়ে, yes, আমরা নিজেদের মধ্যে একটা কনফিডেন্স লেভেল আপনি আমাদের কোভিড-19 চ্যালেঞ্জের মধ্যে পিপিই এর চ্যালেঞ্জটা কিন্তু অন্যতম ছিল। ডেইলি এখন একটা ডেইলি পার্ট অফ লাইফ হয়ে গেছে। তবে আমার মনে হয় কিছুটা হলেও আমরা কোভিড-19 এর ভয়টা অনেকটাই জয় করতে পেরেছি। Yes, we do have nightmares and when we sleep and cry out of fear. But once when we put the PPE kit on, we have to be ready to start. ভেতর থেকে PPE কিটটা পরার পর একটা যে পজিটিভ ভাইব আসে না, যে না আমাদের করতেই হবে, সেটা তুলনা কোনো কিছুর সাথে সেটা আমি সেটা আমিও ফেস করেছি, আর সবাই ফেস করেছে, সেটা তুলনা হয় না। নিজে থেকে একটা পজিটিভ ভাইব চলে আসে কাজটা।
দিনকে আমি প্রথম ফার্স্ট কন্ট্যাক্টে আসি, তো আমি জানতাম না তিনি পজিটিভ। তো তার সেই ডক্টরের যে পেডিট্রিশিয়ান পজিটিভ ছিলেন, তো তার কন্ট্যাক্টে গেলাম, তার পরের দিন জানতে পারলাম হাইলি ডিটেক্টেড তার পজিটিভ আছে। টুনার টেস্ট হচ্ছিল এবং হাই ডিটেকশনটা খুব হাই ছিল। তো শুরুতে আসলে একটু ভয় পেয়ে গেছিলাম। তো তারপরে আমরা টেস্ট করি পজিটিভ ছিলাম। তো সেক্ষেত্রে দেখা যাচ্ছে যে যেটা আমরা কাজ করতে করতে যেটা ফিল করেছি যে এই যে আমরা কি
পেশেন্টরা যখন হাসি মুখে সুস্থ হয়ে যাচ্ছে এবং সেই কনফিডেন্সটা গ্রো করছেন যে তারাও সুস্থ হয়ে সম্পূর্ণভাবে বাড়ি ফিরতে পারে, এটি অন্যতম প্রধান পাওনা আমার এই এক্সপেরিয়েন্স। আর এর পাশাপাশি পার্সোনাল চ্যালেঞ্জের প্রচুর ছিল শুরু থেকে, তাছাড়াও নানা রকম পারিপার্শ্বিক প্রতিকূলতার মধ্যে কাজকে কন্টিনিউ করতে হয়েছে। সেই জন্য এই ব্যারিয়ার্স গুলো কোথাও না কোথাও আমাকে আরো স্ট্রেন দিয়েছে, মেন্টাল স্ট্রেন দিয়েছে।
ভালো অভিজ্ঞতাটাই আমি ভাগ করে নিতে চাই। যেমনই 15ই আগস্ট আমাদের এলাকাতে আমাকে সম্মানিত করা হয়েছে একজন কোভিড ওয়ারিয়র হিসেবে। এটাতে আমি সত্যি ভীষণভাবে আনন্দিত এবং গর্বিত, এবং আমার মনে হয়েছে যে সত্যি আমি কিছু একটা কাজ করতে পেরেছি যেটা আমাদের সমাজের জন্য মানে উপকারী বা ভালো কিছু একটা সত্যি করতে পেরেছি।
যদি বলেন আমার নিজের এক্সপিরিয়েন্স, ইটস অ্যাবসলিউটলি ডিফারেন্ট। কারণ এখন মনে হচ্ছে আই বিকাম স্ট্রঙ্গার, আই অ্যাকোয়ার্ড মোর কনফিডেন্স। আজকে আমার মনে হচ্ছে আমাদের অ্যাজ আ ডায়েটিশিয়ান, অ্যাজ আ হেলথ কেয়ার প্রফেশনাল অনেক অনেক রেসপন্সিবিলিটি। শুধু তাই নয়, যখন হসপিটাল থেকে বাড়ি ফিরছি তখন নিজেকে একদম স্যানিটাইজ করতে হচ্ছে, ভাবতে হচ্ছে আই হ্যাভ মাই ফ্যামিলি। আমারও বাবা মার বয়স হচ্ছে, তাদের ইমিউনিটিটা আমাদের থেকে অনেকটাই কম। তো সেইগুলো, সেই রেসপন্সিবিলিটিস গুলো অনেকটা এসেছে। এবং কিছু জিনিস তো ডেফিনেটলি খারাপও হচ্ছে, যেমন মানুষ অনেক বেশি প্যানিক করছে। আমি নিজে ফেস করেছি, আমার নেবার্স, আমার নোন অনেকে বলেছে অ্যাজ আ ডায়েটিশিয়ান তোর কি কাজ, মেডিকেল ফ্র্যাটার্নিটিতে তোদের কোনো কাজ নেই। এই যে সবাই ইমিউনিটি নিয়ে কথা বলছে, অথচ অনেকেই বুঝতে চাইছে না যে পেশেন্টের রিকভারির জন্য প্রপার নিউট্রিশন ইন্টারভেনশনটা খুব দরকার।
Every doctors, every dietitians, every healthcare workers are playing a great job in this pandemic situation. My respect for all of you. Thank you.
মানছি সময়টা কঠিন, কঠিন সময়টা এখন আছে, পরবর্তীকালে কিন্তু কঠিন সময়টা থাকবে না, কিন্তু এই সময় আশায় কিন্তু ভরসা হয়ে দাঁড়ায়। আমরাও কিন্তু ব্যতিক্রম নই। সাধারণ জনজীবন এখন বিপর্যস্ত করোনা ভাইরাসের জায়গায়, এবং যারা বাড়িতে রয়েছে বা আমরা যারা উদ্যোগের সাথে এগিয়ে চলেছি করোনা ভাইরাসকে জয় করবো, সকলের মনে কিন্তু এখন একটাই আশা, আমরা করবো জয় একদিন। আমরা করবো জয় একদিন।
আমি এটা আমার ডিউটি করা, কাজটা করা। আর এই কাজ করে যদি আমি সত্যি আপনাদের কিছু হেল্প করতে পারছি, যদি সত্যি পেশেন্ট সুস্থ হয়ে বাড়ি ফিরতে পারছি, এখানেই আমার
ভালো থাকবেন, সুস্থ থাকবেন, এবং প্রত্যেকটা ফ্রন্টলাইন ওয়ার্কার, স্পেশালি ডক্টরস এন্ড সিস্টার্স, যারা প্রত্যেকটা দিন প্রত্যেকটা মুহূর্তে করোনার সাথে লড়াই করে চলেছেন, আমাদের কমিউনিটি রক্ষা করছে, তো তাদের জন্য একটা বিগ স্যালিউট। ভালো থাকবেন, সুস্থ থাকবেন। অনেকটা আশা যে উই শ্যাল ওভারকাম। আমরা খুব তাড়াতাড়ি এই করোনা যুদ্ধে জয়ী হব। আমরা সবাই একসাথে থাকব এবং আমরা সবাই পুষ্টিতে থাকব, একে অন্যের সাথে ভালো থাকবো, ভালো রাখার চেষ্টা করব। সো থ্যাংক ইউ সো মাচ, সবাই ভালো থাকবেন।
নিজের নিউট্রিশন এবং নিজের শরীরের খেয়াল রাখুন এবং নিজের ফ্যামিলির খেয়াল রাখুন, এবং চেষ্টা করুন ম্যাক্সিমাম কাজটাকে বাড়ি থেকেই রেগুলেট করার। সুস্থ থাকুন, ভালো থাকুন। থ্যাংক ইউ।
আর আমরা সবাই কিন্তু একদিন না একদিন এই যুদ্ধটা জয় করতে পারবোই, এটা আমাদের মনে বিশ্বাসটা থাকা উচিত। নমস্কার, সবাই ভালো থাকবেন, সুস্থ থাকবেন।
Thank you, sir, Professor Calder. I welcome you. We will start at 5:30.
Can you hear me okay?
Yes, yes, you are audible.
Okay. Okay, I just need to check out the screen sharing.
Yes. The present now button at the right hand side lower corner.
Ah, got it. Thank you.
Yes. Yes, absolutely okay.
Great.
Anna, you may start. Oh, someone has presented. Shahid Hussain Khan. Please.
Participants, it's a request that you don't present your screen. Shall we start, Pubalidi?
Yes, we can start. It is 5:30.
Greetings. It's my pleasure to welcome you all to the 7th day of the International Webinar Series organized to celebrate the Poshan Month. For the last 6 days, we have been experiencing an enormous load of splendid and informative lectures by various eminent speakers from national and international institutes in the field of food sciences and nutrition. As we come to the last day of this webinar series, we have two eminent speakers who will captivate our attention towards the two most important topics like omega-3 fatty acids and microbiome towards healthy living. Now, without further delay, I call up Amrita, our research scholar, to introduce our speaker, Professor Calder. Welcome you, sir. Amrita?
Yes, ma'am, I'm here. Greetings and a very warm welcome to all of you on the 7th and final day of the week-long international webinar series.
Amrita, 1 minute. Shehran Parveen, please stop
Okay.
presenting.
Participants, it's a request that you don't present your screen and put off your mics when the speakers are speaking, please.
Snehara Parveen, please. Please stop the presentation. Snehara Parveen, please stop the presentation. Snehara Parveen.
Organizers, it's my earnest request that in case of this kind of disturbance, please remove the person from the webinar.
Snehara Parveen, it's a request to you that you don't present your screen. I think the organizers have to decide now.
Amrita, carry on. I'm removing him.
Thank you, ma'am. Thank you very much. Greetings and a very warm welcome to all of you on the 7th and final day of the week-long international webinar series organized by the Department of Home Science, University of Calcutta on the occasion of National Nutrition Month 2020. Without further ado, we move on to today's first lecture that will be delivered by our distinguished speaker, Professor Philip Calder. Professor Philip Calder is currently serving as a professor of nutritional immunology within medicine at the University of Southampton, England. His work aims to understand how nutrition affects the functioning of the human body. He has broad research interest in nutritional modulation of immunity, inflammation, and cardiometabolic disease risk. Much of his work has been devoted to explore the metabolism and functionality of fatty acids with an emphasis on the roles of omega-3 fatty acids. He has over 500 scientific publications and he has listed as a highly cited researcher. He is a fellow of the Royal Society for Biology and of the Association for Nutrition. Professor Calder has shown that the fatty acids like eicosapentaenoic acid and docosahexaenoic acid are able to inhibit partly a number of aspects of inflammation, including leukocyte chemotaxis adhesion molecule expression and leukocyte endothelial adhesive interactions, production of eicosanoids like prostaglandins and leukotrienes from the N6 fatty acid arachidonic acid, production of inflammatory cytokines and T-cell reactivity. We are extremely fortunate to have Professor Calder with us, who will be speaking on the topic omega-3 fatty acids and health across the life course, my journey. Participants, you are requested to mute yourselves, not to turn on your camera, and not to present your screen. You can type your questions in the chat box. The moment has finally arrived for one of the most awaited lecture. Welcome, sir. The platform is eagerly waiting for you.
Okay, thank you. I'm just searching for, I had my screen up before. Now let me see. Okay, this was working just fine before. Can you see my presentation?
No, sir.
No, sir.
First, you have to open the presentation.
Yeah, it is open. Sorry. This was working just fine before.
Shima Singh, can you please turn off your camera? Yes. Thank you very much.
Okay, we're in business now. I think you can see my screen okay.
Yes, sir, it is visible.
Okay, great. Sorry about that. So thanks so much for asking me to present to you as part of this important Nutrition Week. So a lot of my research has been on omega-3 fatty acids in relation to inflammation and immune function. But I decided today, rather than going deeply into that topic, which only some of you might be interested in, to actually talk about omega-3 fatty acids and human health right across the life course. And I'll illustrate the point that omega-3 fatty acids are really important to human health by pulling out papers from our own contribution to the research, as well, of course, as important papers from other people. So I'm going to start my presentation by showing you the first paper I published on fatty acids. Now, this paper was published in 1990. I had actually published a number of papers on some other topics before then, but this is my first full paper on fatty acids. And what I did in this bit of research was I looked at the effect of different fatty acids on a process called phagocytosis using cells called macrophages in culture. So macrophages are one of the cells of the immune and inflammatory systems, and phagocytosis is the process by which macrophages remove debris or damaged material and also remove bacteria. So the process is shown on the right here. So the macrophage membrane actually changes its configuration so that it can engulf a particle like an entire bacterium and then take that particle up into the macrophage and destroy it. So obviously the membrane is quite important in this process. So what I did was I incubated macrophages with different fatty acids and then looked at their ability to carry out this process of phagocytosis. So shown on the x-axis here is a summation of the fatty acid composition of the membrane of macrophages. And the membrane composition was changed by incubating the cells with different fatty acids. And what you see quite clearly is the ability of these cells to carry out phagocytosis varies according to the index of unsaturation of the membrane. In other words, it's something to do with the fatty acid composition of the membrane. And sometimes the cells work better, and other times they don't work so good. And this is somehow related to something to do with the fatty acid composition. A little while after that, I studied T lymphocytes, again in cell culture. And again, the fatty acid composition of these cells was changed by incubating them with different fatty acids. So this is a summation of the fatty acid composition. And here I measured the fluidity of the membrane. That is how liquid the membrane is. And what you see is there's a relationship between fatty acid composition and membrane fluidity. In the previous slide, there was a relationship between fatty acid composition and cell function. So this leads to the very simple idea that the fatty acid composition of the membrane affects the physical nature of the membrane, what I've called fluidity, and in turn, this affects function. So this has been the starting point of my research over more than 30 years now. Of course, it's more complex than this, as you'll see as we go along. So what I want to do today is introduce you to omega-3 fatty acids. What are these fatty acids? Where do they come from and how much do people eat? What happens if you increase the blood and tissue levels of omega-3 fatty acids through increasing intake? Say something about how these fatty acids work, then talk about some different elements of health and disease. I'm going to start with inflammation, then move on to heart disease, and then talk about the role of omega-3 fatty acids in early life, from pregnancy, in fact, pre-pregnancy, all the way through to childhood illness. If I have time, I'll mention some 21st century issues and what the solutions to those might be. So omega-3 fatty acids are polyunsaturated fatty acids. That means they have several double bonds in their hydrocarbon chain. There are several families of polyunsaturated fatty acids, but the main ones are the omega-6 and the omega-3 family. And of course, today I'm mainly focusing on omega-3 fatty acids. These terms, omega-6 and omega-3, actually relate to a structural characteristic in the fatty acid, and that is the position of the double bond counted from the carbon at this end of the fatty acid chain. So in an omega-6 fatty acid, this double bond is on carbon number 6, and in an omega-3 fatty acid, this double bond is on carbon number 3. 1, 2, 3. So there's nothing magic about this term omega. It's simply a chemical descriptor of the structure of the fatty acid. So omega-3 fatty acids are a family of unsaturated fatty acids. We call them long-chain, highly unsaturated fatty acids. As I've mentioned, the name comes from the position of the double bond closest to the methyl terminus of the fatty acid chain. There are several members of the family, including alpha-linolenic acid, eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA. As you'll see, the different members of the family have different dietary sources and actually different biological roles, but they are metabolically related to one another. In fact, this is the metabolic pathway relating the different omega-3 fatty acids to one another. So the simplest fatty acid in the family is this one here, alpha-linolenic acid. It has 18 carbons and 3 double bonds. Today I'm going to be talking a lot about EPA and DHA. EPA has 20 carbons, 5 double bonds. DHA has 22 carbons, 6 double bonds. So it's evident to get from alpha-linolenic acid to EPA, you have to make the carbon chain longer and you have to insert some more double bonds. So the desaturase enzymes are responsible for inserting new double bonds, and the elongase enzymes are responsible for making the chain longer. So we have delta-6 desaturase, that's the rate-limiting enzyme in the pathway, that adds a double bond to alpha-linolenic acid to produce stereotonic acid. So we go from an 18 carbon 3 double bonded fatty acid to an 18 carbon 4 double bonded fatty acid. This is then elongated by the addition of 2 carbons down this end of the chain. So we maintain the omega-3 bond at the other end of the chain. Then there's another desaturation with the insertion of a 5th double bond to give us EPA. Then there's a complicated pathway to go from EPA to DHA that involves elongation, of course, and the insertion of an additional double bond. It's important at this stage to say that these same enzymatic reactions occur in the omega-6 fatty acid family. So this is another version of the pathway that I just showed you, going from alpha-linolenic acid all the way to DHA. And then this is the analogous omega-6 pathway, starting at linoleic acid, the simplest omega-6 fatty acid, and going first to arachidonic acid, which sits in the same place in the pathway as EPA, and then on to osbond acid, which sits in the same place in the omega-6 pathway as DHA. So the same enzymes are used in both of these pathways, and therefore the omega-6 and omega-3 families are competing with one another for these enzymes. This is a direct competition between the different substrates. So this brings us to the idea of balance, the need to balance up the supply of linoleic and alpha-linolenic acids so that these pathways occur as they should to maintain optimal function and health. And of course, an imbalance of the pathway would mean maybe we didn't have optimal function or optimal health. And then further down the pathway, you might imagine there's some balancing between the different analogs. So for example, the balance between arachidonic acid and EPA could be important in terms of inflammation, immune function, blood clotting, and so on. So I've told you about the pathway, and I've told you the last steps going from EPA to DHA are rather complicated. And it turns out that this pathway actually doesn't work very well in humans, or at least in most humans who have been studied. There is some conversion of alpha-linolenic acid to EPA, but the conversion on all the way to DHA is quite poor. In fact, there are studies showing inability of some humans to produce DHA all on their own. And what this means probably is preformed dietary sources of EPA and DHA are really important. So let's look at the dietary sources of these different omega-3 fatty acids. So firstly, alpha-linolenic acid is produced in plants. It's an essential fatty acid. Humans can't make it. So it's a plant-derived fatty acid, and therefore it's found in some seeds, like flax seeds or linseeds, some nuts, particularly walnuts, and some vegetable oils, like soybean oil and rapeseed oil. In contrast, EPA and DHA are mainly found in seafood, especially in what we call fatty or oily fish. EPA and DHA are also found in supplements, so what you might call fish oils, cod liver oil, also in some algal oils, in krill oil, which some of you might have heard of, and there are also concentrated pharmaceutical preparations of EPA and DHA that can be used in particular medical conditions. DHA is also found in human breast milk, and I'll come back to that point later on. So alpha-linolenic acid comes from plants, EPA and DHA comes mainly from seafood or from supplements. If we look at the typical dietary intakes of the different omega-6 and omega-3 fatty acids that I've mentioned, you'll see they're quite different. So in Western populations, linoleic acid, which again comes from plant sources, intakes are around 10 grams per day. Alpha-linolenic acid intakes are somewhere between 0.5 and 2 grams per day. So I've said around about 1 gram a day. So the balance between linoleic and alpha-linolenic acid in the diet of many people in the West is 10:1. Intake of arachidonic acid, which comes from meat and eggs, is about 0.5 gram a day. And intake of EPA and DHA is really small, 0.1 gram per day, 100 milligrams a day, maybe even less in many people. But of course, it will be higher than this in fish eaters and people using supplements. So the different members of these families are present in the diet in different amounts. But in general, the diets have an abundance of omega-6 fatty acids and a low amount of omega-3 fatty acids. This is reflected in blood lipids and cell membranes and what is in tissues in humans. So this is data from one of our own studies where we looked at the fatty acid composition of immune cells from the blood of healthy volunteers. This is the contribution of these fatty acids to all of the fatty acids in the membrane. So the total, of course, would be 100%. And what we see, so these are the average values from a group of human volunteers, 10% linoleic acid, 20% arachidonic acid, and 2% of this fatty acid, which is an intermediate in the pathway. So 30% or more of omega-6 fatty acids. And then we see almost no alpha-linolenic acid, 0.5% of EPA, and 2.5% of DHA. So around about 3% of omega-3 fatty acids. So just as the diet contains about 10 times more omega-6 than omega-3 fatty acids, so these cell membranes contain about 10 times more omega-6 than omega-3 fatty acids. So there must be some sort of relationship, and I'll come back to that when I show you some experimental data later on, linking dietary intake and cell membrane composition. Now, I've shown you here the immune cells from people's blood. Now, if we analyze EPA or DHA in different compartments in the body, different pools in the body, we see different amounts of the fatty acids. So this is the contribution of EPA to the total fatty acids in different blood lipids, triglycerides, phospholipids, cholesterol esters, free fatty acids, in red blood cells, in different white cells, in part of the brain, in the retina in the eye, in heart muscle, skeletal muscle, and so on. So generally speaking, EPA is present at rather low amounts, somewhere between 0.1 and 1.5% of total fatty acids. So there's not much EPA in most body compartments. This is the same data for DHA. So immediately you see a different scale. For EPA, the scale went up to 1.6. For DHA, the scale goes up to 14. That's because there are some body compartments that contain large amounts of DHA. So for example, the cerebral cortex contains about 8% of its fatty acids as DHA. Retina, 12% of fatty acids is DHA. Sperm, 10% of fatty acids is DHA. This suggests that probably DHA is very important in those places, in the brain, in the eye, in sperm. And I'll come back to some of those points later on. If we put those two sets of data together, so EPA is in blue, DHA is in the browny red color here, we see there's always much more DHA than EPA wherever we look in the body. And maybe that means that DHA is functionally more important than EPA. That would be one interpretation of these data. So it could mean that DHA intake, DHA supply, blood levels of DHA are really, really important to function of some cells and tissues, maybe particularly the brain, the eye, sperm, and maybe some other places. So I've already mentioned the sources of EPA and DHA, seafood, especially oily fish, and different supplemental forms. Let's look at the fatty acid composition of fish now. So this is the amounts of EPA, DHA, and the intermediate docosapentaenoic acid, DPA, in different types of fish. And I've drawn a blue line here separating the fish listed at the top and the fish listed at the bottom of the table here. And the reason is the fish at the top are what we call fatty or oily fish. They contain large amounts of EPA and DHA. The fish at the bottom are called lean fish or white fish. They contain much lower amounts of EPA and DHA, although they are still good sources of EPA and DHA compared to other foods. But what you can see is one meal, one serving of something like canned sardines or salmon could give people 1, 2, maybe even 3 grams of EPA plus DHA per day. This is actually EPA plus DPA plus DHA. Let's say 1 to 3 grams. In contrast, one meal of something like cod will give people about 1/10 of that amount, 300 milligrams. But remember, the typical daily intake is only 100 milligrams anyway. So even white fish can make a significant contribution to intake of these fatty acids. It's important to note in passing that the presence of these fatty acids in fish is actually because of their progress up the food chain. In fact, marine microalgae are the source of EPA and DHA for the food chain and are simply passed up the food chain to fish. So the primary source of EPA and DHA on Earth is microalgae. And that's really important because algae could become an important source of EPA and DHA if fish are limiting. Let's now look at supplements. And here I show you the amounts of EPA and DHA from 1 gram of any of these oils. And usually in a supplement, the capsules contain 1 gram of oil. There are smaller ones that contain 0.5 gram of oil. So this is the amount in 1 gram of oil. So you see something like cod liver oil or krill oil will give people 200 milligrams of these fatty acids. There are concentrates. There are special algal oils that are used in the infant formula industry that contain a lot of DHA and not much EPA. And then there are these pharmaceutical preparations that I mentioned before that contain 80 to 90% of EPA plus DHA. So just keep in mind, if there's 200 milligrams of EPA plus DHA in a cod liver oil capsule, there's 800 milligrams of other fatty acids, saturated fatty acids, monounsaturated fatty acids, and so on. So fish oil is not equivalent to EPA plus DHA. It's simply a good source of EPA plus DHA. Then finally at the bottom, you see flaxseed oil doesn't contain EPA and DHA. It contains the plant alpha-linolenic acid. So you must be very careful in discriminating supplements sold as sources of omega-3 fatty acids between those that provide the plant omega-3 fatty acid and those that provide the fish omega-3 fatty acids. Because as you will see, these are very different from one another biologically. So the point of this slide is to show you that someone consuming a normal diet with low amounts of EPA and DHA can actually greatly increase their intake of EPA and DHA by using omega-3 supplements, either standard supplements or concentrates, or by consuming fish. And of course, consuming oily fish gives a huge increment in EPA plus DHA intake on a daily basis. These other two bars are prescription grade concentrates of omega-3s, where you could prescribe up to 4 grams of these for triglyceride lowering, which would give people an enormous intake of these fatty acids. So we can increase intake by eating fish or by using supplements. And we know if that happens, the amounts of these fatty acids in people's blood, in blood cells, and in many tissues like liver, heart, and skeletal muscle increases. That effect is dose dependent. In other words, the more you take on board, the more ends up in these different compartments. It's time dependent. So there's a period of time over which this happens. And it's tissue dependent, by which I mean these different compartments change to different extents. Before showing you some data on this, we just need to think about the whole body handling of fatty acids. So omega-3 fatty acids in the diet, as with other fatty acids, are mainly present as triglycerides. These must be digested and absorbed in the gut. Then they make their way into the bloodstream. So here I've called this a transport pool, because these fatty acids have been carried in triglycerides, phospholipids, cholesterol esters to different tissues. They could go to the adipose tissue and be stored. They could go to the liver and be metabolized, or they could go to other cells and tissues, immune cells, heart cells, brain cells even, where they're incorporated into membranes. So I've called these functional pools. So you have transport pools, functional pools, storage pools, and metabolic pools of these fatty acids. There are many different pools, many different compartments, different chemical forms. I've mentioned triglycerides, phospholipids, cholesterol esters. And within one compartment like blood, there may be many pools: red blood cells, white blood cells, platelets, the different lipids that are components of lipoproteins, free fatty acids, and so on. And as I've already indicated to you, the content of EPA and DHA varies in these different compartments and pools. So what is the relationship between increased intake of EPA and DHA and the amount of those fatty acids in different pools in the human body? So this is a simple study where the researchers simply measured people's fish intake, and then they measured the amounts of EPA and DHA in red blood cells. And the combined percentage of EPA and DHA is termed omega-3 index. So this is a readout of omega-3 status. And what this simply shows is the more frequently people eat fish, the higher is the content of EPA and DHA in red blood cells. And this will also prevail elsewhere in the body. So fish eating is a way to increase omega-3 status. I've already said that. We've done quite a few studies using supplements where you can do randomized controlled trials to look at changes in physiology, changes in biochemistry, changes in functionality. So this is one of our studies where we looked at the incorporation of EPA on the left and DHA on the right into immune cells from people's blood over the course of 12 weeks of supplementation. So these volunteers took omega-3 supplements every day for 12 weeks, and we took blood at 4, 8, and 12 weeks. And what you can see is EPA is enriched by 4 weeks and doesn't really go much higher after that. DHA is enriched after 4 weeks and doesn't really go much higher after that. So these fatty acids get into immune cells in humans, certainly within 4 weeks. Then we stopped supplementation and we took another blood sample after another 8 weeks. And what you see is the EPA has returned to normal, or has returned to baseline. So in other words, EPA gets in easily while you're supplementing, but if you stop supplementing, it comes out easily. DHA gets in. If you stop supplementing, actually it comes out more slowly. This observation has been repeated several times over, and not just for immune cells, but also for platelets and red blood cells. And again, I think maybe this points to the fact that DHA is functionally more important than EPA, and therefore there are mechanisms for cells to retain DHA when its supply is limiting. Nevertheless, the point of this is there's a time course for these fatty acids getting in. This is the dose-response relationship from another one of our studies where we gave volunteers different amounts of EPA for 12 weeks, up to 4 grams per day. So this is a very high dose. And this is the change in the amount of EPA in the blood immune cells. And what we demonstrated was this linear dose-response relationship up to quite a high dose. So in other words, the more of this fatty acid you consume, the greater the amount you end up with, in this case, in immune cells. And again, this has been shown for other sorts of cells. Now, we were very interested in drilling down into the detail of these time courses and dose-response relationships. So we did a study where we had 200 healthy volunteers and we supplemented them with different doses of omega-3 supplements every day for 1 year. So this is a very long supplementation study using a high number of volunteers who were in 4 different groups. There was a placebo group who were consuming capsules or supplements without omega-3s, and then there were 3 groups who were consuming supplements that provided different amounts of omega-3 fatty acids. They provided both EPA, which is at the top, and DHA, which is at the bottom. And then this is the amounts of those fatty acids in phospholipids in the blood. But we saw the same picture for other blood lipids, for red blood cells, for white blood cells, for platelets, and so on. And that is, there's a clear dose-response relationship. So this is a particular dose, this is twice that dose, and this is 4 times that dose. So there's a very nice dose-response relationship, and you see that EPA peaks, gets to the new level at all doses within a few weeks. DHA takes a little bit longer. So there's a pattern of a dose-response relationship of EPA getting in quickly compared to DHA, but both of them getting in eventually. Here I plot the dose-response relationship between these 4 different doses and the increment in total EPA plus DHA, but the data are the same for EPA and DHA alone, showing this dose-response relationship into the different blood lipids, into white cells, and into red cells. So there's a clear dose-response relationship, and we also identified the time course of the incorporation here. Now remember, the volume, the size of a cell membrane is set at a particular limit. So cell membranes can't grow and contract the way that adipose tissue can grow and contract. So as EPA and DHA are incorporated into cell membranes, they must replace other fatty acids. So indeed, their incorporation is at the expense of other fatty acids, and one of the fatty acids whose content decreases is the omega-6 fatty acid arachidonic acid. So you could see this as a rebalancing of membrane polyunsaturated fatty acids, driving out arachidonic acid, which remember is present at very high levels, and allowing incorporation of EPA and DHA. Some other fatty acids also change when EPA and DHA are incorporated, including linoleic and oleic acids. This shows that this linear dose response incorporation of EPA is linked to a linear dose response reduction in arachidonic acid content. As I say, you could view this as a rebalancing of membrane fatty acid composition. So why is this all important? Why am I making these points so clearly and consistently to you? And the answer is about the link between intake of these fatty acids, the biological effect, and the clinical outcome. And I try to capture this in my mechanism slide here. So what I've shown you is by increased oral intake of EPA and DHA, we end up with more EPA and DHA in the blood. We also end up with more EPA and DHA in cell membranes. I showed you right at the beginning that fatty acid composition of cell membranes can influence the fluidity, that is the physical makeup of the membranes. Part of the importance of that is cells assemble proteins together in their membranes as part of signaling events. These assemblies of proteins are called rafts. The assembly of rafts requires proteins to move in the plane of the membrane, and that is influenced by the fluidity of the membrane. So through changing membrane composition, we can actually affect the assembly of these signaling platforms. Another element to this is that chemical mediators are produced from cell membranes, including from the fatty acid components. Things like eicosanoids, that's prostaglandins, leukotrienes, and so on. Resolvins, which are pro-resolving mediators that I'm going to come back to later on. There are also some receptors that seem to be rather specific for DHA, which means DHA can act in a hormone-like manner on some cell types. So all of these membrane-mediated effects, and remember right at the beginning, I stressed my early view that the membrane was really important, influence the signals that cells are producing, the intracellular signals and the extracellular signals. Now, intracellularly, that means that cell and tissue responses can change, including the pattern of gene expression. This is really important when fatty acids control metabolism or inflammation, for example. What this means is cells behave differently. So you've impacted on physiology and maybe on pathology as well. What I'm going to show you is through this general cellular and molecular mechanism, EPA and DHA act to improve physiology, to improve health, decrease disease risk, and give better clinical outcome. Essentially, they set a balance in favor of health and optimal function compared to disease and impaired function. So through those mechanisms, EPA and DHA can affect visual function, cognitive function, metabolism, inflammation, immunity, oxidative stress, coagulation, the function of many organs, wound healing, and many other things, insulin sensitivity, and so on. So in other words, increased EPA and DHA in the diet alters fatty acid composition of cell membranes, more EPA and DHA, less arachidonic acid. This improves cell and tissue phenotype. This improves health or clinical outcome. This is my hypothesis anyway. So in other words, having more EPA and DHA in the blood, in cells and in tissues is associated with better health. So this leads on to the next part of my talk, which is to look at omega-3s in human health across the life course. And I'm not going to start at the beginning and go to the end. I'm actually going to dive in with things to do with adults and older people and then go back to early life. I'm going to start with inflammation because inflammation is really important in many biological processes. So normally, inflammation is helpful. Inflammation is physiological. It helps with our immune defenses. So normally the inflammatory response is triggered, it goes on for a period of time, and then there are signals to turn inflammation off. This is called resolution of inflammation. I'll come back to that shortly. So normally, inflammation is self-limiting. But if this process of resolution fails, then inflammation continues and becomes chronic. This can lead to tissue damage, pathology, and disease. And there are many diseases which are linked to inflammation. And of course, there are many anti-inflammatory drugs that are used in lots of different clinical settings. Turns out EPA and DHA have many anti-inflammatory mechanisms. I don't want to go into all of these in details, but I summarize some of them here. As you've seen, EPA and DHA reduce the amount of arachidonic acid in cell membrane phospholipids. Arachidonic acid is a precursor for prostaglandins and leukotrienes involved in inflammation. So this fire here is the fire of inflammation. EPA and DHA also interfere with the assembly of these signaling platforms that I already mentioned, rafts, in inflammatory cells. And what that means is early inflammatory signaling is disrupted. There's a very important transcription factor involved in inflammation called nuclear factor kappa B. This is a protein which controls the expression of genes involved in inflammation. By upsetting this or interfering with this signaling process around lipid rafts, nuclear factor kappa B is not activated. There's another mechanism by which omega-3s can interfere with nuclear factor kappa B activation. Nevertheless, omega-3s reduce NF-kappa B activation. That means it can't move to the nucleus and it can't activate the genes that encode all of these different proteins, cytokines, chemokines, adhesion molecules, the enzyme COX-2 that produces eicosanoids, matrix metalloproteinases that degrade tissues. So there are multiple sites where omega-3 fatty acids impair the inflammatory process. In other words, they are anti-inflammatory. If you want to know more about the anti-inflammatory mechanisms of omega-3s, I point you in the direction of two reviews here, one from 2015 and one from 2017, where I talk a lot about the anti-inflammatory mechanisms of these fatty acids and the clinical effects of those mechanisms. Just show you a little bit of data that we've recently published looking at endothelial cell inflammatory responses. So endothelial cells are the cells that line the blood vessel walls. If they're inflamed, that's bad news for the individual because that endothelial inflammation can drive atherosclerosis, the buildup of fatty material in the blood vessel walls that ultimately leads to heart disease. So what Ella Baker did here was she incubated endothelial cells with EPA or DHA, and then she stimulated them with this inflammatory cytokine tumor necrosis factor alpha. And when you stimulate the cells, they produce large amounts of these other inflammatory cytokines, which are all known to be involved in atherosclerosis. Now, if she pre-incubated the cells with EPA or DHA, you see the ability to produce all of these inflammatory mediators was decreased, often in a fairly concentration-dependent way, and often with an indication that DHA was more potent than EPA. So again, this brings out the idea of dose-response relationships and maybe that DHA is more biologically active than EPA, which is one of my recurring themes. She also looked at the activation of nuclear factor kappa B, this transcription factor that I've been talking about, and she showed that DHA really inhibits NF-kappa B activation. And then this is the protein COX-2, which is the prostaglandin producing protein, decreased by both EPA and DHA, but again by DHA more so than EPA. I mentioned that this process of resolution of inflammation is really important. So what I've shown you is the lighting the fire of inflammation, and then I indicated at some stage you have to turn off inflammation. So the fire brigade has to come and put out the fire. So about 20 years ago, people got interested in what were the chemicals, what were the signals that promoted resolution of inflammation. So they used animal models to identify these chemicals involved in resolution. And they found out that just like lipid mediators are involved in the initiation and propagation of inflammation, so lipid mediators are also involved in the resolution of inflammation. And they identified new families of lipid mediators that hadn't been described before. And they gave these names resolvins, because they resolved inflammation, protectins, and morescins. And together, these have been termed specialized pro-resolution mediators, or SPMs. It turns out these SPMs are produced from the omega-3 fatty acids EPA and DHA. And I outline the pathways here. So EPA is converted via the cyclooxygenase enzyme in the presence of aspirin particularly to the E-series resolvins. DHA is converted again by the COX-2 enzyme in the presence of aspirin to the D-series resolvins and to protectin D1, and via 12-lipoxygenase to the morescins. So EPA and DHA are the source of the chemicals that turn off inflammation. I think this is really a remarkable role for these fatty acids in human biology. So the idea is you have triggers for acute inflammation, and normally, as I've told you, that inflammation will resolve. And omega-3 fatty acid derived specialized pro-resolving mediators, the resolvins, protectins, and morescins, are involved in that resolution. If they're not present, or they're not present in sufficient quantities, acute inflammation could become chronic, and ultimately that causes pathology, causes disease. Now, experiments have shown, this is experiments in animal models, have shown that you can use these omega-3 derived pro-resolving mediators to rescue chronic inflammation, to resolve ongoing chronic inflammation. And then, of course, the resolution leads to tissue repair, healing, and homeostasis. So these fatty acid derived mediators are really important physiologically in controlling resolution of inflammation, and they could be involved therapeutically in treating chronic inflammation. In fact, omega-3s can be used to treat chronic inflammation, and it may be that they act through generating these pro-resolving mediators. So what I've done now to my scheme is I've added the generation of SPMs, specialized pro-resolving mediators, to the scheme here, and I've shown them as the fire brigade. So all of these other mechanisms mean that the fire that's lit is not as strong as it might be otherwise. So you've got a weaker inflammatory response, but also you've got the initiation of the resolution of inflammation, the fire brigade. The one inflammatory condition I want to talk about is rheumatoid arthritis, and that's because this is really a prototypical inflammatory condition where inflammation is destroying the joints of the individual. So inflammatory cells make their way to the joints, they become activated, they produce the sort of chemicals I've been talking about, prostaglandins, leukotrienes, inflammatory cytokines, chemokines, matrix metalloproteinases, and so on. These destroy the joint. And of course, the individual ends up like this: swollen joints, tender joints, pain, loss of function. This condition is treated with anti-inflammatory drugs. But it turns out omega-3 fatty acids can be very useful in inflammation as well. So there have been a lot of randomized controlled trials done of omega-3s and rheumatoid arthritis. One of the common outcomes in those trials is pain, because of course this condition is very painful. These are two systematic reviews. One of them is also a meta-analysis, both published in 2017, that show that high-dose EPA and DHA decrease pain in people with arthritis. They have other clinical benefits as well. I'll skip the next slide and move on to my last slide about inflammation. And this is inflammation in the context of aging. But there's actually a process called inflammaging, and that's because inflammatory markers have been shown to be elevated in older people. So aging is a state of chronic low-grade inflammation. And it's believed this inflammation associated with aging is very important in terms of predisposing to some of the non-communicable diseases of aging: cardiovascular disease, insulin resistance and diabetes, cognitive decline, loss of lean mass and mobility, immune decline and increased susceptibility to infections, and many cancers. So the inflammation associated with aging seems to be important in all of these conditions. And what I've told you is that omega-3 fatty acids are anti-inflammatory. Therefore, they could be very useful in preventing inflammaging and maybe even in combating inflammaging through the mechanisms that I've talked about. So their health consequences could be very broad in the population. I think prevention is better than treatment. So getting these fatty acids in early and preventing or slowing the development of inflammaging is probably going to be a better strategy than treating existing inflammaging. Although, of course, when you have people with these conditions, you need to treat them. So one of the conditions I mentioned there was vascular disease. So I want to talk now about coronary heart disease and omega-3 fatty acids. This has been studied for decades now. This is data from a cohort study, which some of you will be familiar with, the Nurses' Health Study. What they did here was they measured EPA and DHA intake when the nurses were disease-free, and then they followed them for a period of time, and they looked for the emergence of coronary heart disease, mortality due to coronary heart disease, and having a myocardial infarction or heart attack. And for all of these outcomes, the finding was the same. The higher the intake of EPA and DHA, the lower the risk of these cardiovascular or coronary outcomes, with a risk reduction of about 50%. And remember, you would have a high intake of these fatty acids from high fish consumption or use of supplements compared to a low intake from low fish consumption and not using supplements. This is a similar set of data, this time from the Physicians' Health Study, where they took blood from the physicians when they were disease-free. They measured omega-3s in the blood of those individuals. They followed them for a period of time, and the outcome here is sudden death. So this is death from myocardial infarction, unanticipated and rapid death as a result of a heart attack. And again, what they showed here was a significant inverse relationship between the amount of EPA and DHA in the blood and risk of sudden death, even after or even with adjusting for many other factors that might be involved, with a risk reduction of something like 80% in those with the highest amount of omega-3s in the blood compared to the lowest amount. And again, you would have a high amount of omega-3s in the blood through high oily fish consumption or intake or use of supplements. So those sorts of cohort studies have been subjected to some systematic reviews and meta-analyses. So this is a meta-analysis from Chowdhury, published in 2014. So what Chowdhury did was they gathered all of the data from cohort studies where people had looked at dietary fatty acid intake in the context of what they call coronary outcomes. And that's because the studies would report different things: incidence of heart disease, dying of MI, surgery for heart disease, or whatever. So they gathered the studies looking at these coronary outcomes. Some of these studies had looked at dietary long-chain omega-3 fatty acid intake, that is EPA and DHA. In fact, there were 16 studies looking at EPA and DHA involving over 400,000 people. And what the researchers showed was that having high intake of EPA plus DHA was associated with lower risk of coronary outcomes compared to having low intake. So this is gathering together cohort data looking at intake of these fatty acids, showing an association with lower risk of coronary outcomes. They also looked at blood levels of the fatty acids. Here they could discriminate EPA, DHA. Some studies reported EPA plus DHA, and some studies reported the intermediate docosapentaenoic acid. Again, they showed having high amounts of these omega-3s in the blood was associated with a lower risk of coronary outcomes compared to having low levels in the blood. So high dietary intakes compared to low intakes and high blood levels compared to low blood levels are associated inversely with coronary outcomes. There are more recent cohort studies showing these same sorts of things. So I think there's robust recent evidence that EPA and DHA decrease the risk of developing heart disease. Of course, these are association studies, not randomized controlled trials. But the question is, how do omega-3s lower the risk of coronary heart disease? So we have to look at the risk factors for cardiovascular disease, and I list those risk factors here. And what I've done is put a tick beside the risk factors that are improved by omega-3 fatty acids. So these fatty acids actually have multiple effects on risk factors, or they have effects on multiple risk factors. So they improve the risk factor profile, and that probably underlies this effect on reducing risk of the disease. This is a meta-analysis published in 2018, looking at randomized controlled trials of EPA and DHA and cardiovascular risk factors. So here we see effects on triglycerides and other blood lipids, on blood pressure, heart rate, and inflammation. And we see the number of studies that have been performed. So for some of these risk factors, there are hundreds of studies that have been performed in humans, the effect size, and the statistical significance. So omega-3 fatty acids decrease triglycerides, they raise HDL cholesterol, they decrease blood pressure a little bit, they decrease heart rate, and they increase heart rate variability, which is a good thing, and they decrease inflammation. And this particular meta-analysis involving 6 studies, they didn't improve vascular function, but there is actually another meta-analysis including more studies that shows an improvement of vascular function with omega-3s. So I think this is good evidence that these fatty acids have effects on multiple risk factors that probably explain this protective effect. If we look at the development of heart disease over time, what I've shown is 2 different trajectories here. One is this trajectory here, where a person is on an accelerated pathway to the disease, and then there's another slower trajectory here. And what these data indicate is there's a preventative effect, a reduction in risk of disease. So omega-3s could be used through daily intake through the diet or with supplements to have a preventative effect to put people on a different track. Let's look actually now at people at risk and whether omega-3s can be used as a treatment. So I have to go back 20 years now to show you this landmark trial in 11,000 people who had had a heart attack but survived, put into a randomized controlled trial of omega-3 fatty acids at 1 gram per day versus placebo, and followed for 3.5 years. And the outcome was mortality. These are the relative risk reductions of mortality related to cardiovascular disease in individuals consuming omega-3s compared to consuming placebo. So these were all highly statistically significant reductions. Other causes of death, like cancer and so on, were not affected with omega-3s. So this was a specific effect on cardiovascular disease. A trial of 18,000 people in Japan using 1.8 grams of pure EPA per day for 5 years, again showed a reduction in coronary events that could be needing surgery for coronary disease, having a heart attack, dying from heart disease. There was a reduction overall in the omega-3 group compared to the control group. And in this trial, everybody was consuming statins. So the control group was on statins alone. The omega-3 group was on EPA plus statins. And you see this reduction in risk of coronary events that was seen in that study. So those studies were published in 1999 and 2007. In the period 2008 to 2010, there are actually studies published showing that omega-3s had no effect. These studies were criticized for various reasons, one of them being the small size, the low omega-3 dose that was used, or the low event rate that occurred. This is a meta-analysis published around that time. looking at omega-3s versus control in randomized controlled trials and cardiovascular outcomes. This shows a tendency towards mortality advantage of omega-3 fatty acids, but only 1 out of 4 of these outcomes, in fact 1 out of 5, was significant. The others were just approaching significance. And this led to the view that maybe omega-3s were not so useful as treatments for cardiovascular disease after all. About a year ago, in fact less than a year ago, this trial was published in New England Journal of Medicine. It's called Reduce It. It was a trial in over 8,000 individuals. They had cardiovascular disease or diabetes or other risk factors, and they were all taking statins, and they had high fasting triglycerides. They were given 4 grams of EPA for 4.9 years on average. This is the outcome. This is actually the same graph, just shown twice using different axes, so we can look at this one here, showing the outcome, which is cardiovascular death, non-fatal heart attack, non-fatal stroke, needing surgery, or developing unstable angina as a composite combined outcome, showing the likelihood of this outcome was significantly reduced in those individuals consuming EPA compared to placebo. This is a forest plot showing all of the different outcomes reported in that study. So this is favoring EPA on this side. This is favoring placebo on this side. This is the primary outcome that I just showed you. Then you see omega-3s were beneficial towards the secondary outcome and to different components of the primary and secondary outcomes. Having an MI, needing surgery, having a stroke, death from any cardiovascular cause, and so on. So I think this is a trial which indicates at a high dose, omega-3 fatty acids actually can be used to prevent quite significant outcomes, including mortality. Just recently, there was a meta-analysis published of these trials here. It includes the Italian study that I showed you, the Japanese study that I showed you and Reduce It, as well as a number of other studies. The conclusion of this meta-analysis is that omega-3s lower the risk for myocardial infarction, heart disease death, heart disease, cardiovascular death, even when the Reduce It trial isn't included, but the effects were greater when Reduce It was included. And these researchers concluded that the risk reductions seem to be related to the dose of omega-3s, which makes sense if you think of the early part of my talk. So I've lost the lines on the graph here, sorry, but I showed you that omega-3s could be used for prevention. Now I'm showing you data that suggests they maybe can be used at high dose for treatment. I'm going to skip these next few slides and move forward. But if you want to get an update on omega-3s and cardiovascular disease, this is a review that I published just a few months ago that brings together the cohort and randomized controlled trial data and brings together the historic and the recent studies. So I've talked about inflammation, I've talked about heart disease. I want to now go back to early in the life course and talk about some things to do with infants and children. So I'm going to start right at the beginning, which is fertility and conception. So I already told you that sperm has a high concentration of DHA. DHA is the major polyunsaturated fatty acid in sperm. It's found in both the head and the tail, although the head contains more DHA than the tail. DHA is very important for tail flexibility and sperm swimming. And the head of the sperm, the DHA content of the head of the sperm is very important for the fusion of the sperm with the egg. It's been identified that dietary fatty acids can affect sperm fatty acid composition. In other words, if a man consumes more DHA, his sperm contains more DHA. And actually the function of the sperm, the quality of the sperm, has been improved in some trials through that sort of strategy. This has been studied, the functional significance of this has been studied a little bit in people undergoing in vitro fertilization. What's been demonstrated is the Mediterranean diet, including fish, has been shown to be associated with greater success of pregnancy in vitro fertilization. Now, that could be due to an effect on the quality of the sperm, or the quality of the egg, or the quality of both of those things. Embryo quality has been associated with alpha-linolenic acid, DHA, and total omega-3 intake. So quality of sperm, quality of the embryo, and likelihood of becoming pregnant, at least in IVF, could all be promoted by omega-3 fatty acids. So we were a bit interested in this idea that omega-3 fatty acids could impact on the sperm, the egg, and the embryo. So recently we completed a randomized controlled trial in couples undergoing in vitro fertilization. So as I say, this was a randomized controlled trial. For each day for 6 weeks prior to egg retrieval and the in vitro fertilization, both the man and the woman consumed a fruit-based drink that provided 2 grams of EPA plus DHA and 10 micrograms of vitamin D. The control group consumed the same drink, but without the omega-3s and vitamin D added to the drink. And the main outcome was embryo quality. This wasn't a study about becoming pregnant or anything like that. It was too small. It was actually about embryo quality. Now, in IVF, of course, the sperm and the egg are put together in a test tube. There's a fertilization process, and then the fertilized egg and then the early embryo are grown in an incubator. And the embryologists actually track what's going on, and they decide at a particular stage, here we can put this embryo back into the lady. So there are all of these checkpoints where you can identify the progress of the development of the embryo and embryo quality. And we were interested in some of these markers of embryo quality that are listed on the right here. The meaning of these don't matter today, but these are indicators of embryo quality that are used clinically. So here are the markers that we used, and these are the numbers, which aren't important, but what we found was that some of these measures of embryo quality were improved in the omega-3 group compared to the control group. So this is the first data from a randomized controlled trial that show that omega-3 fatty acids can improve embryo quality in humans. And I think that's really quite remarkable. Let's move on through pregnancy and into early infancy. Let's think firstly about the brain and about the eye. These are very rich in fat, particularly the brain. I already showed you that the brain and the retina of the eye contain a lot of DHA. They have a high DHA content. The synapses, the gray matter, the retina, very rich in DHA. Some of these chemicals produced from DHA, like the protectins and resolvins that I mentioned before, could be very important in brain development. DHA also has identified roles in the structure and function of membranes in the brain and in the eye. Now remember, the human growth spurt, brain growth spurt, starts about week 28 of gestation and continues up until age about 1 year. And there's massive DHA incorporation into the human brain late in gestation and following birth. So that's shown here. This is birth, and this is going out to 7 years, and this is brain weight. So you see the brain starts growing late in pregnancy and continues up growing for about a year or so after birth. And then this is accretion of DHA in the brain. This is milligrams per gram of brain. So this, if you look at total milligrams of DHA in the brain, you have to multiply these two together. So there's a very steep incline in brain DHA content late in pregnancy and in the first 1 to 2 years of age. And that accumulation of DHA in the brain and in the eye is functionally important. So that accumulation is happening during pregnancy and in early infancy. So let's look in pregnancy. On this side of the placenta, we have the mother's circulation. On this side, we have the fetal circulation. And here's the placenta here. This is a scheme which shows arachidonic acid and DHA in different places on the mother's side and on the fetus's side. So let's just look at blood lipids and DHA. So these are the contributions of DHA to blood lipids on the mother's side, and these are on the fetal side. So look at phospholipids, 6.6 on the fetal side, 1.7 on the mother's side. Triglycerides, 0.4 on the mother's side, 1.6 on the fetal side. So the fetus is actually concentrating DHA. The fetus is dragging DHA out of its mother. What this means is maternal intake of DHA and endogenous synthesis of DHA is really, really important during pregnancy, both for the mother herself, but also to supply this huge demand for DHA by the growing fetus. These are our own data, just to make the same point. We measured DHA on the maternal side and in cord blood, so on the fetal side, and again we see this concentration on the fetal side of somewhere between 2 to 4-fold levels. So the fetus is really concentrating DHA out of the mother. Now, what people have shown is that maternal and fetal blood and blood cell DHA is increased by maternal DHA intake from supplements. There are lots of studies of that. In other words, if a mother takes omega-3 supplements, her own DHA levels go up, but also the fetal DHA levels go up. Likewise, maternal and fetal blood and blood cell DHA can be increased by fish consumption, but there are not many studies of that. We conducted a study in pregnant women, giving them salmon twice a week from week 19 of pregnancy until they gave birth. We took 3 blood samples during pregnancy, and we took cord blood at birth. These are, in this slide, EPA, and in the next slide, DHA, in the maternal circulation, in the control group in blue, and in the women taking salmon in green. So you see EPA depletion in the control group, and that this doesn't occur. In fact, EPA goes up in the salmon group. So eating fish during pregnancy, twice a week during pregnancy, stops the pregnancy-induced decline in blood EPA. There's also a decline in blood DHA during pregnancy for the reasons I just indicated. The fetus is sucking DHA out of the mother across the placenta. But if women eat fish, this decline doesn't occur. In fact, there's an increase in DHA. So again, consuming fish during pregnancy can stop the pregnancy-induced decline in DHA. And then this is what happens in the umbilical cord blood. So this is on the fetal side. The fetal bloodstream has more EPA and more DHA if the mother has been eating salmon twice a week during pregnancy than in the control group where they weren't eating salmon. So those studies show that a woman can supply more DHA and also EPA, but DHA is really important, to the fetus by taking omega-3 supplements or by eating more fish during pregnancy. Let's move on to the newborn infant. Breast milk, of course, contains fat. Fat contributes up to 60% of breast milk calories. It has a high content of the saturated palmitic acid, the monounsaturated oleic acid. There's DHA in breast milk at about 0.32% of fatty acids, although there is a range, and that range is determined by maternal diet. So DHA in breast milk is determined or responds to maternal diet. Women who take omega-3 supplements during pregnancy or lactation have more DHA in their milk. Breast milk can provide 110 milligrams per day to an infant. Breast milk DHA is lower in vegans than in omnivores, so we need to think about that. As I mentioned, breast milk DHA can be increased by increased DHA intake from supplement in the mothers, but can also be increased by fish consumption. And again, I show you data from our study where we gave salmon to some women during pregnancy. Remember, they stopped taking the salmon once they gave birth. This is the DHA content in their breast milk 1, 5, 14, and 28 days after giving birth, in the control group in gray and the salmon group in blue. And you see DHA content even out to 1 month after stopping eating salmon is higher in the salmon group than in the control group. Why is this important? Well, it's been shown that breast milk DHA and infant DHA are positively associated with visual function and with mental development. So there's an association between having higher DHA and having better visual and mental function. There's been interest in what is the impact of adding DHA to infant formula. And there are studies showing that DHA in formula or formula with DHA results in better visual and mental development compared to standard formula without DHA. Although not all studies report that, probably because the DHA dose is different in different studies. It may be we need DHA intake going right out to 12 months to have this effect. There are also some long-term effects of early DHA supply in infants. Blood pressure is lower and obesity is lower in some studies in babies or in children who consumed DHA early in life compared to those who didn't. Let's move on in the life course and talk about a couple of other things, allergies and infections. Talk about allergies first. Here we have to see that the eicosanoids, the prostaglandins and leukotrienes produced from arachidonic acid actually have a role in allergic inflammation. They're causative chemical agents involved in allergic inflammation. In addition, prostaglandin E2 controls the immunology that underpins allergic inflammation. So having too much arachidonic acid seems to predispose to and create the manifestations of allergic inflammation. I already alluded to the fact that omega-3 fatty acids decrease arachidonic acid levels in immune cells. In fact, I showed you one piece of data around that. What that means is there's less arachidonic acid to produce all of these chemicals that are involved in allergic inflammation. What that means is there's less prostaglandin E2 to create the immune situation that underpins allergic inflammation. So maybe omega-3 fatty acids could be used to prevent allergy. So there have been trials of this. This is one trial from Sweden. In this trial, women during pregnancy and lactation took fish oil supplements or not in the control group. And then the infants were looked at up to 12 months of age. And the outcomes here were being sensitized to any allergen, being sensitized to egg, having eczema associated with immunoglobulin E, so that's allergic eczema if you like, or having food allergy. And what the researchers showed was the infants at 12 months of age all had lower levels of sensitization, of eczema, and of food allergy compared to those in the control group. So if the mothers consumed omega-3s in pregnancy or lactation, their children were less likely to have allergic outcomes. A high dose of omega-3s was used in that trial. This is a more recent study, again, a rather large study using a fairly large dose of EPA and DHA, where women took fish oil during the second half of pregnancy or they were in a control group. And here the outcome was wheeze or asthma in the children going out to 5 years of age. This trial was just looking at allergic outcomes up to 1 year of age. Here we're looking at asthma going out to 5 years of age. And you see if the children were born to mothers who took omega-3s in pregnancy, they had a reduced risk of developing asthma compared to the children of mothers in the placebo group. So omega-3s in pregnancy seem to reduce allergic disease and asthma in the offspring going out to several years of age. We were interested in this in our salmon in pregnancy study that I've already told you about in the context of allergic outcomes. So one of the things we looked at was the immunology of the umbilical cord blood. So this shows the production of prostaglandin E2, which you'll remember is central to this allergic process, from cells from the cord blood of control pregnancies or salmon pregnancies, the entire group or a particular subgroup that we looked at. That's, the difference between these two groups doesn't matter, but what's clear here is the production of pro-allergic prostaglandin E2 is reduced if the mothers took salmon during pregnancy or ate salmon during pregnancy compared to if they didn't. And if we looked at 2.5 to 3 years, looked at doctor-diagnosed asthma and wheeze, we found that this was significantly reduced or nearly significantly reduced in the children from the salmon group compared to children in the control group. I think the last set of outcomes I want to talk about is infectious disease in school children. I just want to show you two trials that we've been involved in, one done in Thailand and one in South Africa. So in this study, we had 9 to 12-year-old school children in Thailand who every day or 5 days a week for 6 months consumed a milk that was fortified with 200 milligrams of EPA and 1 gram of DHA. And the outcomes here were illness. This is mainly respiratory illness, of course. So the number of children who got sick was significantly, over the 6 months, was significantly reduced if they consumed fish oil compared to placebo. And the number of days that they were sick in those who became sick was decreased by 50% in the fish oil group compared to the placebo group. This is a study in iron deficient South African school children who were given iron or omega-3s or the combination of iron and omega-3s. What we showed was that, again, as we saw in Thailand, omega-3 fatty acids resulted in fewer illness days. The interesting thing was, if these iron deficient children were given iron, they actually showed more respiratory illness than the placebo group. In other words, giving them iron was harmful. I think that's a very interesting observation in the context of treating nutrient deficiencies. Giving them omega-3 fatty acids alone actually had very little effect on the respiratory symptoms, but omega-3 fatty acids prevented the adverse effect of iron on respiratory illness. So this is a very interesting interaction between omega-3s and iron in the context of respiratory disease that we're following up a little bit to try to understand what's going on there. So I've talked about fertility, I've talked about visual and cognitive development in infancy, I've talked about allergic disease and illnesses in childhood, and I've talked about cardiovascular disease or heart disease and inflammation in adulthood. And I think the picture that comes out of this is that omega-3 fatty acids, or EPA and DHA, really promote optimal physiological function, reduce disease risk, and result in health benefits right across the life course. So what I've told you is these fatty acids are metabolically related. Their roles and mechanisms of action have been widely studied. The intakes are usually low, but can be increased through diet, which I think is the preferred option, oily fish, or supplements. That increased intake of EPA and DHA is reflected in higher levels of these fatty acids in blood and in tissues. They act through several mechanisms to improve cell and tissue function in a way that's consistent with improved health across the life course. So the bottom line is eat fish. There's a problem here because fish is not sustainable. Some people, of course, don't eat fish, can't eat fish, don't like fish, but nevertheless, even for those people who like fish, fish is not sustainable and fish stocks are overexploited. So what we have to do, I think, is look for some alternatives to EPA and DHA to achieve the functionality that I've been talking about. So the most obvious question is, well, what about the plant omega-3s? What about alpha-linolenic acid? What about stereotonic acid, the product of delta-6 desaturase? There's been a lot of research in humans looking at alpha-linolenic acid in particular. We did some research in this area some time ago, looking at alpha-linolenic acid and comparing the effects to EPA and DHA. Sure enough, alpha-linolenic acid does have effects in humans, but the effects are much less than the effects seen with EPA and DHA. In other words, the plant alpha-linolenic acid is weak in its biological function compared to EPA and DHA. A few years ago, we reviewed all the literature on alpha-linolenic acid in humans. We identified that probably the main role of alpha-linolenic acid is actually to act as a precursor to EPA, DPA, and DHA. But as I said right at the beginning of my talk, the ability of humans to make DHA seems to be quite poor, although actually it seems to be better in females than males. High intakes of alpha-linolenic acid actually can have functional effects. alpha-linolenic acid can have the same effects of EPA or similar. This seems to be related to alpha-linolenic acid dose, so you need very high intakes, and seems to be related to conversion to EPA. So here we plotted from different studies alpha-linolenic acid intakes that were used and the change in EPA that was seen in blood lipids. And we showed that these are linearly related. In other words, the more alpha-linolenic acid people are eating, the more EPA that appears in their bloodstream and in their cells and tissues. But these are very high intakes. Remember, the typical intake in the West is around 2 grams per day. And of course, we can get much bigger increments in EPA simply by giving people EPA. So maybe there is some benefit in alpha-linolenic acid, but it's not a complete substitute. What about stereotonic acid? Again, there have been human studies of stereotonic acid. We did talk about these in the review that I just showed you. Stereotonic acid can enhance EPA status better than alpha-linolenic acid. That's because it's the product of the rate-limiting step of the omega-3 pathway. Stereotonic acid can mimic the effects of EPA and DHA, probably due to conversion. But again, conversion of stereotonic acid all the way to DHA is low. So there's another option. Now, I told you before that plants don't produce EPA and DHA, but actually you could genetically modify plants to do that. And we've been working with this researcher here, Jonathan Napier. He's a plant biotechnologist. He's genetically modified this organism, this plant, Camelina sativa, to produce an oil. This is the seeds of that plant. The oil from these seeds actually is very similar to fish oil. So it's not like a plant oil, not like the native plant oil. It's actually more like fish oil. And recently, we published the first human papers giving this transgenic oil to humans. And we showed that if we match the EPA and DHA content of the fish oil to this transgenic plant oil, they both behave in the same way. So there is a genetically modified plant alternative to EPA and DHA from fish. So these are the alternatives, I think. Firstly, remember that algae are sources of EPA and DHA. So algae and algal oils are good, sustainable, vegan and vegetarian friendly sources of EPA and DHA. I talked about plant omega-3s, alpha-linolenic acid and stereotonic acid. Maybe they are alternatives, but we probably need high intakes. And there remains this question about their ability to be converted to DHA. I've just talked about GM plants that produce EPA and DHA. This could be part of the future. Another alternative, of course, is to look for non-omega-3 fatty acids that have similar biological effects. We've been involved in that research recently. And then the final way actually is to try to make our endogenous pathway for making EPA and DHA from alpha-linolenic acid work better. One way to do that would be to lower linoleic acid intake, because if you lower linoleic acid intake, you reduce the competition between the omega-6 and omega-3 families, and you promote EPA and maybe even DHA synthesis. The other thing I didn't mention is the enzymes involved in that pathway require trace elements like zinc. And there are data showing that if you improve trace element status, you can improve the endogenous pathway of synthesis. So these are interesting things for the future. So I'll finish here. Thanks very much for your attention. I hope there were some useful and interesting things in there for you. Okay, I'm going to stop sharing now and hopefully return to where we were.
We are mesmerized, sir, with your lecture.
Okay, well, thank you so much.
I hand over the mic to Amrita. She has all the questions. I don't think we have any question. You have given us, enlightened us every aspect of EPA and DHA. And
Yeah.
only one thing I was we worked on RBCs of uncontrolled diabetic patients.
Yes.
And they have a very different sort of RBCs with polygonal shapes under scanning electron microscopy, and they are all rigid membranes. Can EPA and DHA help in reducing this the fluidity of the membrane in case of diabetic patients?
Yeah, so there are some other diseases where the red blood cells take on different shapes and different rigidity, and there is evidence that omega-3s can be useful there. So I think it's worth trying whether you can alter the composition and maybe increase the fluidity so that the membranes become less rigid. I think that would be very worth doing. Yes.
Thank you, sir.
Yeah, I do see there have been some questions, but maybe there's another way to deal with those.
Yes, Amrita will choose the questions and put to you. Amrita.
Thank you, sir. Thank you. Such an informative session and an amazing presentation too. With your permission, I would proceed with the questions.
Oh yeah, go for it.
Thank you, sir. The first question, is there any effect, that is damage or any reaction on DHA during cooking or processing fish, like deep frying or shallow frying, etc.?
Yeah, that is a great question. So the answer is yes. So when you cook fish, particularly oily fish, of course you lose some of the oil. That means you lose some of the omega-3s. But also if you're using high temperature, you will oxidize the omega-3s, and that's not helpful. So I think you have to be very careful with the cooking process. Of course, deep, pan-fried salmon still contains omega-3s, but not as much as maybe salmon prepared in other ways. So you have to be very careful with the cooking process, yes.
Thank you, sir. Next, in mother's milk of Bengalis, we found EPA and DHA content more than many countries. Can it be due to the synthesis of DHA from linolenic acid in lactating mothers at a higher level?
Yeah, I think that's another interesting question. So people have looked at the DHA content of milk taken from women from many different countries, and it's highly variable, obviously within a country, but it's very variable between countries. And some of that relates to the diet in different countries, but I'm sure some of it relates to the metabolism in different countries, which will be affected by all the sorts of things that I've talked about, like alpha-linolenic acid and linoleic acid intake, zinc intake, maybe some other micronutrients, other things in the diet. So I think the observation is very interesting, and is probably, I mean, it's either diet or metabolism, or the combination of the two that is causing
Yes.
that difference. So I think we should look at the diet.
Sir, the Bengalis, we take a lot of fish, and along with it, we take mustard oil, which contains erucic acid, which is a typical fatty acid that is present in Bengali diets. There may be some pathways, which I think that the Bengali mothers, they produce a little more, about 1 gram DHA in their milk. Okay.
Yeah.
Thank you.
Yeah, I think you have the answer. They eat more fish, and also they have some other dietary differences.
Yes.
Yeah, that's interesting.
Next, in the present scenario of COVID-19 all over the world, a brand new concept is rising persistently. The patients died off not only due to the immune deficiency, but also the hyperimmune activation, that is hyperinflammation. Do you think that the EPA-DHA supplementation can be used for mitigating inflammation in future clinical practices? If it is, then how can we determine the dose?
Yeah, so that's another, these are all good questions. So in fact, I have written about this very topic. So I think I wrote one review about nutrition immunity in COVID-19. And in part of that review, I wrote about the possibility that omega-3s could be used in severe COVID-19 for exactly the reason your questioner asked, which is controlling the hyperinflammatory response. No one has done any trials yet, but there are studies in acute respiratory distress syndrome in other settings that show very high doses of omega-3s can be useful. So I think, you know, you have to be careful in these situations, but I think you probably need quite a high dose if you're interested in looking at that, by which I mean, you know, a couple of grams a day.
Okay, sir, you are talking about the high dose. So the next question, what is the upper dose limit up to which EPA DHA supplementation can be increased without any possible adverse effects?
Yeah. So yeah.
Okay, you may proceed, sir.
Yeah. So in experiments, people have used extremely high doses. Okay, and they are doses that wouldn't normally be used in normal practice. There are very few studies showing adverse effects of EPA and DHA, and I think the doses that I have been talking about, which are high hundreds of milligrams to several grams per day, you know, I talked about going up to 4 grams per day, for example, in some studies. There are no adverse effects. Okay, so there are some European bodies that have looked at what the upper limit should be, and usually they come up with something like 5 grams per day, and that's of EPA and DHA, not of oil, right? And remember, often the oil isn't totally pure. So, I mean, I think, you know, if people are interested in doing things, using 2 or 3 grams a day seems a reasonable amount.
2 or 3 grams. Okay, sir. What's your take on the bioaccessibility and bioavailability of EPA DHA from high dose supplementation?
So if we think about healthy people, humans are very good at accessing macronutrients from the diet. Okay, so we're very good at digesting fat, carbohydrate, and protein, and absorbing the products of that digestion. So in healthy people, where omega-3s are consumed as part of a meal, the bioavailability is very high. Okay, if somebody takes supplements without a meal, that can be a problem, because I'm sure you've studied digestion, that you need some fat to promote all the digestive machinery. So if you take supplements without a meal, you don't have the fat load that will promote digestion, and then bioavailability actually can be very low. Quite a few people have shown that now. So if people take supplements, they should really take supplements with a meal. Okay, and if you do that, that's not a problem. Now, of course, if you have somebody who's got some gastrointestinal condition that can affect digestion, absorption, the situation can be different. But in generally healthy people, there isn't a problem so long as omega-3s are part of a meal.
Amrita, I have a question for Professor Calder. We work in nano emulsions of EPA DHA. One of my students have done it. And can it be applied in IBD, that is bowel syndrome, where inflammation is a problem, and we are seeing that it can act with nano formulations of EPA DHA. Can it be done?
Yeah. So maybe I'm not sure. I'm not exactly sure what your preparation is like, but this could be a way of delivering omega-3s to the site of inflammation. So some years ago, so there have been some studies on EPA and DHA and in inflammatory bowel disease. Some of those studies work, some of them don't. But there have been some very good studies where the omega-3 capsules have had a coating, which means they go further down the GI tract, and therefore they release the omega-3s closer to the site of inflammation. So maybe if you have a preparation which does that, that could be very useful.
Yes, thank you, sir.
Yeah, but I'm not really sure what the nature of your preparation is.
Thank you, sir. Next question. I would like to know whether maternal consumption of EPA and DHA can alleviate the developmental neurodegenerative disorder like FASD, fetal alcohol spectrum disorder, etc.
Yeah, so there are actually studies on that, and those studies, both experimentally and I think there are probably some human studies, but the experimental studies suggest this could be an approach. Of course, when people do clinical trials, all sorts of things happen because clinical trials are very different, partly because you can't control everything, and humans are more complicated than rats and mice. So I think, I mean, if people are interested in that, they need to explore the literature that's there, but there are papers on fetal alcohol syndrome and omega-3s.
The next question, how does the homeoviscous adaptation in host cell membrane impact the resident microbiome and pathogens?
Gosh, okay. So I think you're linking with the next talk here. So there are data. So if you're talking about the host rather than the microbiome, there are data. So you have to think about two things. One is any direct effect of the fatty acids on the microbiome, and the other is effects on the microbiome as a result of influencing host immunity, host inflammation, host gut epithelium. So there are data, including in humans, that show omega-3 fatty acids can have effects on the microbiome. There have been some very interesting papers on that. I'm not really a microbiome expert, so I can't offer too much, but certainly there may be changes. And if you think about what I was saying in relation to an earlier question about having preparations of omega-3s that get further down the GI tract, you could have a bigger effect on the microbiome then.
Okay, sir. As consumption of fatty fish of higher trophic level of food chain increases, there is increase in mercury level. So would you suggest to increase consumption of large fatty fish as they are rich in EPA and DHA?
Yeah. I think this is one of the. It's actually related a bit to my point at the end. We're searching for alternatives to EPA and DHA from fish is really important. So as your questioner rightly says, there are some types of fish that are more likely to be contaminated than others. And of course, that will also, there's a geographical perspective to that as well. So I certainly wouldn't want to promote the consumption of contaminated fish, either contaminated with mercury or with things like PCBs. But if we take the contaminants out of the picture, it's obvious that fish consumption can be very healthy, but that would be fish without contaminants. So I think that's a really important question. And, you know, I'm not sure exactly what the answer is, but I think one wouldn't knowingly want to promote contaminated fish.
Amrita, please, our next speaker is
Just waiting patiently.
Thank you, sir, for this amazing lecture once again. Anna ma'am, Anna Lakshmi ma'am, are you here, ma'am?
Yeah, I'm here.
Okay, ma'am. I would request you to express sincere gratitude to Professor Calder on the behalf of our department.
I deem it our proudest privilege to have one of the finest researchers of the world among us and enlightening us with the splendid experience of his life with omega-3 fatty acids. Sir, on the behalf of the organizing committee, I thank you sincerely for your presentation. I am running short of words to express my humble thanks. Please accept our heartiest congratulations and sincere thanks for your remarkable, outstanding presentation at the webinar. Thank you once again, sir.
Okay, it's my pleasure, and good luck for the next lecture.
Thank you, sir.
Thanks very much, everybody.
Thank you, sir.
Now, I would like to invite our next distinguished speaker, Professor Maria Gloria Dominguez Bello. Professor Maria Gloria Dominguez Bello is currently acting as Henry Rutgers Professor of Microbiome and Health in the Department of Biochemistry and Microbiology, Rutgers, the State University of New Jersey, USA. She completed her first postdoctorate research under EU Marie Curie Scholarship in the Institut National de la Recherche Agronomique, France, and Rowett Research Institute, Scotland. Currently, she is serving as a member of the American Academy of Microbiology, American Association for the Advancement of Science, American Anthropological Association, etc. Professor Maria belongs to the editorial board of several journals like MBio, Scientific Reports, etc. Research in her lab focuses on the study based on the co-evolution of the microbiota and host and impacts exerted by the Western lifestyle practices. The aim of the research is to investigate the role of the built environment in microbial transmission, integrating the fields of anthropology and architecture into microbial ecology. Professor Maria contributes over 110 scientific publications in different peer-reviewed journals. She recently published a review describing the role of the microbiome in human development, including evolutionary considerations and the maternal or fetal relationships, contributions to nutrition and growth. There is an enormous opportunity for all of us to hear Professor Maria on the topic microbiome and virome in non-westernized peoples. Participants, you are requested to mute yourselves, not to turn on your camera, and not to present your screen. You can type your question in the chat box. Welcome, madam. The platform is all yours.
Thank you very much, Amrita. Can you hear me well?
Yes, ma'am. You are visible and audible too.
Okay, so good morning, good evening, everybody. I'm going to share my screen. And
ma'am,
can you please tell me if you see it?
Yes,
can you see the first slide?
Yes, ma'am. It is clearly visible.
Okay, wonderful.
Thank you.
All right, well, good day, everybody. It's my pleasure to be here. It's an honor to be invited to your workshop today. I heard a little bit of the last speaker, and the questions and answers certainly were very good and animated and important. I'm going to probably switch gears here and talk about the microbiome and the virome in non-westernized peoples. My research focuses mostly on early life acquisition of bacteria, how babies shape their microbiome, but also how modern life affects that process and ends up in a perturbed microbiome. So I'm going to focus on that aspect. How is the microbiome of people who have not undergone the pressures of urban lifestyle? So in this slide, I depict the tree of life where bacteria were first, then as evolution proceeded, archaea, and then eukaryotes evolved. But with the characteristic that eukaryotes contain the whole tree. They contain archaea and they contain bacteria in their bodies through associations that have co-evolved with them. And then this depicts human evolution. And in every generation, there is transfer of the microbiota from one generation to the others, with the primordial inoculum being from the mother. So there is inheritance of the microbiome. There are lines of inheritance, and the primordial inoculum is maternal. In the old days, humans inhabited the earth, and they lived in very different lifestyles. They conquered the whole world, the whole globe, all the climates, and they live different traditional lifestyles. But today, we are losing that cultural diversity to become a single lifestyle, a single way to live, heavily reliant on technology, technological agriculture, medicine, technology to preserve foods. And this has a lot of implications, and I'm going to focus on the implications for the microbiome. So as we urbanize and industrialized, we move from this traditional, I'm depicting here the house, to an intermediate and then more urban lifestyle. And we know for some time the association between the microbiome and urbanization. On the left panel here is the paper we contributed with Jeff Gordon, the Yatsunenko paper in 2012, where you can see on the y-axis is the observed bacteria, the different bacteria observed, and age. And these are three populations. This was the first study that compared geographically outside a single country. And Malawans are in red, Amerindians, these are indigenous people from Venezuela that live in a rural area, and then the United States. And what you can see is that everybody's born with very low diversity. And as they grow up, diversity increases. But the blue stops growing in diversity before the red and the green. So that was the first time we observed directly the association between urban level and a decrease. The higher the urban level, the lower the diversity of the microbiota in the gut. These results have been reconfirmed many times by now. On the right panel, we see that the Bacteroides is more dominant in the US than in the other two sites, Malawi and Amerindians, that have more Prevotella and other bacteria. So the increase in Bacteroides is one of the markers of urbanization. We know the association seems to be with lifestyle because Malawans are in Africa and Amerindians are in America. So it's not geographical, but probably a lifestyle. This is a little more complex graph, but what I want you to see, the red line here, these are different countries in a principal coordinate analysis 1, in the component 1, and the x-axis is age. So what it shows, as you can see in the red line, which are traditional peoples, is that as age advances, the structure of the community, which is represented by the position in PC1, changes in one direction, whereas in the industrialized countries, it changes differently. You have the reference there, is Fragiadakis paper, and you have the different microbes that increase or decrease in the gray urban or in the red traditional countries. And again, markers of traditional, microbial markers of traditional peoples are the Prevotella, as I said before, Succinibibrios, Spirochetas, Paraprevotellas. This is the most striking figure that I can show you, and it's probably the most important that I'll show you today, because it shows again the principal coordinate analysis 1 position. So what we did is we gather all the data of the world and plotted in what space do they occupy in PC1, in the PCO1. And what happened was an ordination that is a spontaneous ordering of the microbiotas of the world that are deposited, the 16 acids deposited in banks, in gene banks. And when you see the ordering, it's a spontaneous ordering by lifestyle. On the left side of the PC1, you have the Venezuelan Yanomami. This was a work we did with first contact Amerindian peoples in Venezuela. Then followed by Burkina Faso, Peru, Tanzania, so South American and African communities that live in jungle savanna. Then as you move right, these intermediate microbiotas belong to people that are rural. Malawi, Cameroon, Venezuela, Amerindians that are living rural conditions, not in the jungle. Papua New Guinea, Bantu. And then on the right is the urban cloud, starting with Japan and China on the left, and then ending with US, Canada, and Ireland. But certainly even Japan microbiotas that have been published really cluster with the urban cloud. So this is striking because it shows that there is a strong association between urbanization, lifestyle, regardless of the continent, and structure of the gut microbiome. The differences are so striking between jungle, examples of which you see in these photos on the right-hand side, jungle and urban, that we went and decided to do a much narrower urban exposure gradient. Our exposure gradient was in the rainforest village where there is no access by road. You have to fly there. It's 4 hours from Caracas in Venezuela. But you have the community on the bottom is the landing strip. And so that community receives doctors and scientists, ecologists. So they have high urban contact, higher than the other two communities, which are really traditional. And you can see it in the architecture. The bottom community has a traditional hat, but already has houses with the windows, zinc roof, as we know them in the cities. Whereas the other communities are entirely made up of natural materials. To go to these communities is a long walk or river or both, and what we did was we flew by helicopters. Helicopters are in the area because these are areas of river blindness. And there are health programs from the WHO that send helicopters to treat people because they are trying to eradicate onchocerciasis, which is river blindness. So we went there, and that's an area we have been working for some years now. So this is the gut bacterial microbiota diversity and urban exposure, where the urban exposure is low in red or medium in green, and then we sample the doctors that were there, and that will be blue, and they were all adults. So this is by years of age. So what you see is that at any age period, the microbiota in red is higher than the microbiota in green. Low exposure is higher than medium. So you don't really need to change entirely the lifestyle and the diet, because the landing strip community still has the same lifestyle. They don't have a market to buy food. They grow their own food. They use more medicine because they are exposed to mostly medicines from doctors, and a little of the transculturation is already enough to decrease the diversity. You see in adults the trend of decreasing diversity with urban exposure. We did an experiment that we also published in this paper in the bottom of the microbiota diversity of the villagers and the people who go there and stay there for 3 weeks or a month and eat their diet and live their lifestyle. So what we observe in the principal coordinate analysis here, we observe that PC1 separates the indigenous microbiota, gut microbiota, from the visitors. Within the visitors, we observe that eating entirely their diet, number one, do not make the microbiota substantially more similar to the indigenous people microbiota. The dots don't get closer in PC1. But there were enormous changes, especially in some people, in the visitors' microbiota due to the change of diet, most likely. Very high fiber diet, a lot of cassava, fruits, almost entirely vegetarian diet, although there was fish and once in a while game animals, hunting. So your microbiota, if you radically change your diet, will change, but not necessarily will become, at least in this short period of time of 3 weeks, will not become more similar to the structure of the microbiota of the people living there and in that lifestyle, eating that diet for a long time. We also have studied viruses in a gradient that is the same gradient I just told you about. So landing strip, jungle, village versus the most remote villages in the jungle, and Caracas as an urban center in the same country. Of course, in this case, you are introducing the ethnicity difference. In Caracas, the majority of people are mestizo, mixed. In the jungle, they are only Amerindian people. The only non-indigenous people that live in the jungle are a few of the missioners in the mission towns. So this is the description of the communities, and these were focusing children. So we ask, what are the children carrying in terms of viruses in a gradient that goes from A, urban, Caracas, B, the community of the landing strip, C and D, two of the communities deep in the jungle. This is the virus distribution in each children in sites as we decrease urban exposure from A to D. And what you see is that each virus, which is depicted on the y-axis, is represented in a color, and the size of the sphere represents the size of the viral nucleic acid detected. So what we see is that there are less viruses in the urban site than in the other sites. And there are some viruses that the urban site entirely lacks. We can see, so there is a viral, lower viral diversity as you increase urban settings. There are a lot more dots, smaller dots in village D and C than in urban site A. So we urban people have more of fewer viruses. This is the distribution of viruses that are unknown in the children. And what you can see is in the urban side, in the city, there are only basically 2 unknown viruses we couldn't classify. But look at village D. It's really remarkable. The majority of the viruses are unknown. These are the unknown. These are the total known. So there are almost more unknown virus than known. We also study the vaginal microbiome, and we are particularly interested because the vaginal microbiome is the first pioneer source of bacteria that colonize a newborn. So we ask the same question. If we take a gradient of exposure from very low in the jungle to medium to the town, and in this case, the blue it's a small town, but where there is a market and there are houses and roads and traffic lights. It's not a big city, but it's an urban rural setting. Daniela Vargas, my student, did her PhD asking this question, and in the context of also vaginal health, because Amerindian women, the first cause of death in the jungle is HPV cervical cancer. So we wanted to understand the diversity, the distribution of diversity in the town, in more intermediate communities, and in the jungle, because we know that in the jungle there is such a high incidence of cervical cancer, which happens at the global level as well. So she compared Amerindians that live in the jungle, in the intermediate rural town, or in the town. And she had a reference group of mestizo also living in the town. Mestizos don't live in the jungle. So she extracted the DNA, and HPV was detected and classified by type. So the vaginal microbiota does change with urbanization. Her power wasn't great, but there are, if you compare Amerindians and mestizo, we can see the Amerindians are red, the mestizos are blue. The structure separates, although after corrections, the differences were not significant. The major reason was the presence of higher Prevotella and Mobiluncus in mestizos, and higher Peptonephilus and Brevibacterium in Amerindians. Most interestingly, we show on the left side the HPV diversity by urbanization, low, medium, high, or mestizo. And this is the sample size. So this is a rarefaction curve. So what we see is that as we move from low urban to high urban level, there is an increase in the diversity of human papilloma viruses in the vagina. So this is remarkable, because that means the Amerindians, who suffer significantly a lot of cervical cancer, are harboring a lower diversity of HPV. We can see this in the this is a dispersion. So we have in the low urban level, we have lower dispersion of the data than in mestizo, but in mestizos, the diversity is much higher, more different types of HPV. We know more than 100 HPV types. So the prevalence also it has a tendency to increase that is not significant, but on the right graph, we can see that the prevalence is higher of viruses that are only high-risk viruses in relation to the low-risk viruses. And that this happens in Amerindians. So Amerindians have low total diversity, but the few viruses they have are all bad, are all highly virulent. Whereas in mestizos, they have a mixture of high virulence and low virulence viruses. Is an increase in the diversity that is by the non-virulent HPVs. You can see that better in this heat map, where HPV 18 and 39, which are two of the high virulence viruses, are very high in the low urban group, less in the high urban group or in the mestizos. There is a decrease with urbanization. This has important implications because the vaccine has not been introduced in the Amerindian villages yet. And that's something we are proposing to do because precisely the vaccines against HPV are against the most virulent viruses. What are the implications of these changes in the vaginal microbiota with urbanization? What are the implications for the next generation since the babies receive that primordial inoculum? And what happens if you change it? Those are open questions. How does bacterial microbiota alters HPV infection or viral infection? And this doesn't have to be necessarily only vaginal, but also gut. What's the effect of the vaccine introduction, and how does that affect the virulence in general? So we know that urbanization, among other things, mean use of antimicrobials and change of diet, among many other things. So antimicrobials are extremely important because thanks to them is that we have controlled or we can control infectious diseases in densely populated urban sites. So antimicrobials are the key to have antimicrobial sanitation. Antibiotics are the key to control infectious diseases in dense areas, but they are also affecting the microbiota. The main questions are, what microbiome functions are we losing with the loss of diversity? And is this loss associated with the health issues that are typically for urban societies? What are the health issues of urban societies? Well, we know that as infectious diseases are controlled, thank you to in high part to hygiene, sanitation, and antibiotics and medicine, what we observe is a correlated increase in immune disorders and metabolic disorders. So we know that asthma, type 1 diabetes, obesity, multiple sclerosis, Crohn's, and other arise only in urban industrialized societies. So this is the big challenge for the world now. What we are doing is trading diseases. We are trading diseases that are curable for diseases that are incurable, and that the people live the whole life with these diseases. So if microbes are involved, what we propose, and we show that in a perspective in science, what we reason is if industrialization is degrading the gut microbiota diversity, and in black, depicted in black here, and that is associated with an increase in modern diseases, can we then restore the microbiota diversity and revert the disease trend? So for that, because the traditional peoples of the world are disappearing, we are proposing a global microbiota vault to conserve the future health of humanity. We are proposing a global nonprofit initiative where countries collect their own, especially countries that have high microbiota diversity, which tend to be the non-industrialized. Those are the ones who have this wealth. They collect the entire world, help characterizing that, and again, local collections can decide with whom collaborate, and in the future be able to restore. We are proposing a vault that represents both the network of local collections and a central collection, which is in a neutral country, preferably in the Arctic, in the North Pole, and for that, we started an effort to form the team, the global team. This is the pioneer team of the microbiota vault. We could get the support of 2 Nobel Prizes, very remarkable people in the field of the microbiome, people from foundations. We published the feasibility study, and you can see it in the microbiotavault.org. And this is all inspired, you may know, of the seed vault in Svalbard. We are following their inspirational example. What they did is agriculture is killing the natural diversity of seeds, of plants, with a lot of parallelisms to the microbiota. Agriculture, industrialized agriculture is doing to the natural diversity of plant what sanitation and antibiotics is doing to the microbiota. So we...
excuse me, madam, excuse me, someone has presented. Please, Dishani, please stop presenting.
Yes, this is my last slide, actually.
No. Someone has presented. Please, Dishani, please stop presenting.
Dishani
well, that was my last slide. So
no, ma'am. Someone else has presented her screen, so we are not able to see your presentation.
I have already finished my presentation. This is Maria Dominguez.
Dishani Das.
Please, can you present once more?
Ma'am, can you present your last slide once more?
Who, myself, Maria Dominguez?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Oh, okay. So this was my last slide. This is my... So I stop. This is my previous to last. I was saying that we were following the steps of the seed vault, and with that, I thank you so much for your attendance. And this is the team with whom I have worked for years, very closely on this aspect of anthropology of human microbes. And with that, I'm happy to take questions, if there is time.
We have enough time. Amrita?
Yes, ma'am. With permission of Professor Maria, I would like to begin with the questions.
Absolutely. Please go
Thank
ahead.
you, ma'am. The first question is, why and how does the host have different immune responses to commensal bacteria and pathogens?
So that's a very active field of research now. The short answer is we don't know why, but we do know that the immune system is trained both by environmental antigens and also by the own microbes. And in reality, the first exposure that the immune system has to foreign components happens during birth. And that's why I say that the mom's microbes are pioneer. Those are the first microbes that will colonize the baby, and the baby will tolerate them. And informing, so the immune system is allowing those babies to colonize, not attacking those first microbes. But what happens if those first microbes are altered? What happens in C-section born babies who don't receive that first primordial inoculum, but they are receiving environmental microbes, mostly skin microbes floating in the operating room? The immune system is getting a different setting, a different programming. So we know that perturbations in early life antibiotics in early life during the window of development severely alter the immune system. And in adulthood, those people will react different to different antigens. So that's something we want to understand. And there are lots of work being done using models of disease as well, type 1 diabetes, obesity, colitis, even autism and Alzheimer's might be related to that programming of the microbiome development affecting the immune system development.
Thank you, ma'am. Now, why there is so much less studies on human virome in contrast to that of human bacteriome? In the human microbiome, which are more prevalent in terms of number and function, bacteria or viruses?
Yes, that's a very good point. And I think the reason is because it's easier to study bacteria because we know more of bacteria. We know less of other microbes, and the most complex of all, as in the question is implicit, are the viruses. There are relatively few people doing viromes. I think it's going to grow, and we need that, but it's very little what we understand. We have a lot of viruses that live on us without causing disease. We don't understand the ecology. Are they protecting us against bad viruses? Are we affecting the good viruses with practices? We simply don't know that. And I think it's because it's a much more complex, technically complex, but also we know very little.
Okay, ma'am. How does human virome changes with diseased condition of body?
We don't know. I haven't, our virome study was very descriptive. As you could see, these are all healthy people, healthy kids and healthy adults. I'm sure it changes. And again, ill people take medication and that would also change further. So that's a good question. And the answer is, I don't think we know.
Next question. Are bacteriophages present in the human virome? If yes, how bacteria survive in their presence?
So that's a very good question, because that's precisely the ecology of bacterial populations is greatly shaped by bacteriophages. And it has been studied in the gut. I don't think it has been studied in any other colonized body site, in the skin, in the nose, in the mouth, in the vagina, but it has been studied by a few groups in the gut. And the bacterial viruses are shaping really the structure of bacterial populations. And it's not only diet. So it's a very complicated ecological system that we are only starting to understand.
Next, does the human microbiome and virome interact with each other? Can they live amiably in the human system?
So we humans have our own viruses, and our microbes have their own viruses. But our microbes interact with our cells. They are talking all the time, especially to the immune system. So that means that our microbiome may be altering our immune responses, including to viruses. But again, this is a moving field, which is advancing now. The microbiome is more or less 20 years old to human knowledge. And we started studying the easier part, which is bacteria, but certainly viruses are also interacting in the system, in this complex system. So there are relatively few publications.
Amrita, one question from me.
Yes, I'm sure.
Professor Gloria, can the products of these bacteria in the gut, can they affect the brain? Because we have now the nutrient sensing. The word nutrient sensing is coming up. And can these products have some effect? And as gut is called the second brain, so I think they may have some role.
Yes, I think you are right. That's another question that is being researched now. There seems to be metabolites from the gut microbial fermentation that are anti-inflammatory, but are also affecting the brain. And that's why there is a whole area of people studying microbiome and autism, or microbiome and brain integrity. The answer is it seems like, and that's an active field of research right now.
Thank
Next
you.
question. Does human microbiome depend, sorry ma'am, just a minute. Does human microbiome depend on the variety of food consumption among vegetarian, non-vegetarian or vegan people?
Absolutely. Diet is one, it's not the only one, but it's one of the selective factors that alter the diversity, as you can imagine. So every time we eat, we are also feeding our microbes in more or less extent. If you don't eat fibers, if you eat a 100% degradable diet, you starve your hindgut microbes, and you undergo severe pain. So those experiments were done in humans, in astronauts. They at some point they thought, if we can make the astronauts not having to go to the bathroom, we don't need a bathroom in the shuttle. So they tried to feed astronauts with 100% absorbable diets, and they couldn't do it because they got sick. So you need to feed your microbes, but the way urban people tend to feed their microbes is very poorly. We eat in urban societies, one of the characteristics is a very low quantity and low diversity of fibers, or polysaccharides that are undegradable to human enzymes. The human milk has a lot of compounds that are not degraded by the baby but by the bacteria, and they booster those bacteria. But the same with our diversity of plants, greens and fruits. Those will provide different substrates for a diversity of bacteria. So we think that part of the high diversity in the jungle that we observe in the Amazon is sustained by high diversity of plant fibers.
Thank you, ma'am. Do you think healthy microbial transplantation can be implemented to ameliorate malnutrition and other diseases?
Yes, that's a very good and important question. I know the work of Jeff Gordon on malnutrition, and he has shown that certainly the microbiome is responsible for the different responses to renourishing children. So some kids are refractory to have a nutrition restored, and it is because of their abnormal microbiome. So there is a lot of promising technology and methods to restore the nutrition of malnourished children, considering the microbiome. We still don't know which are the key microbes, but I think we will. Let me tell you that again, malnutrition, undernutrition is still a problem in some parts of the world, and it will be and especially with the perspective of the global climate. There will be droughts and which will lead to undernutrition. And we have that problem ahead and present, but we also have the huge problem of overnutrition, which affects all developing countries and industrialized countries. So we have the two problems in the two extremes of the nutrition gradient.
Okay, ma'am. Diet and habitat can manipulate gut microbes. How does this affect other aspects of healthier lifestyle?
I don't think I understand the question.
Okay, ma'am. I repeat again. Diet and habitat can manipulate gut microbes. How does this affect other aspects of healthier lifestyle?
Other aspects of healthy lifestyle.
Yes, ma'am.
So I guess, diet, environment, lifestyle, what you eat. And we know now that exercise affects the microbiome as well. Stress affects the microbiome. So it's not only the diet, it's also the way you live. It's not only the way you eat, but also the way you live and your lifestyle, including exercising, including stress. So it's an extremely complex system. And we are trying to disentangle and understand what is the effect of each one of the factors. And we know the answer is yes, they affect. And what is the optimal? Well, probably the optimal is to live a good life where you are happy, where you exercise and rest enough and sleep enough. I mean, all we have associated with healthy diet. What we need to understand is why. What is the mechanism?
Thank you, ma'am. How have bioinformatics accelerated the growth of this field of study? In future, is the study in this field possible without bioinformatics?
I don't think that's possible I mean, bioinformatics is crucial because that's the tool. It's like those are the glasses through which we see the microbiome. These are all letters and you have to make sense of them. And the bioinformatic tools are absolutely essential to be able to study these systems. We are not, it's an evolving field, and bioinformatic tools are improving and need to be improving. There will be no end because after we understand better the bacteria, then we need to understand what's the interaction with viruses or with fungi. So and with the human expression, which is also, you can measure as transcriptome. So it's extremely complex and we are not there yet. We need bioinformaticians definitely to help us make sense of such a complicated system.
Thank you, ma'am. Next question. How and who are prone to hypothalamus pituitary axis in case of recurrent microbes attack, mainly by HPV?
That's a very specific question, and the answer is I don't know. Probably somebody is working on that. HPVs and the brain and the pituitary. The answer is I don't know.
Okay, ma'am. Next question. Would you suggest paleo diet to restore gut microbial diversity?
Can you repeat the question?
Yes, I'm sure. Would you suggest paleo diet to restore gut microbial diversity? Paleo diet.
So diet and microbiome diversity restoration. So the answer is diet is crucial because imagine you have a low microbiome diversity and you increase it via ingesting the microbes you lack. If you don't provide them with the substrates in the diet, they will die off again. What I think in the future is that what I think will happen in the future is that we will understand the system enough to restore it every time we need. For example, we need an antibiotic because we have an infection. If we understand enough the system, we could take the antibiotic and follow by restoring the unintended consequences of the antibiotic, restoring the good microbes that were affected. So definitely, and again, you need to be aware of providing the microbes with the substrates. So, and that comes with the diet, absolutely.
Thank you, ma'am. The next question, and probably the last question. Instead of doing fecal testing to check gut microbiome, is there any alternative method?
There are new techniques. There are capsules that are that test on the way down the gut. It's an expensive technology and not widely available yet. Again, they will sample the lumen content at different distances of the mouth. What we also would need and would be nice to have is an endoscopic capsule that samples also the tissue. I think that's technically possible, but we don't have it there yet. These are less invasive access to our gut microbiome.
Sorry, ma'am, there is another question. Effects of pre and probiotics on gut microbiome.
So probiotics and prebiotics are a field that was really grew outside the scientific world first. And that poses a problem because basically our shelves in the supermarkets and pharmacies and healthy stores are filled with probiotics and prebiotics that have not been tested enough scientifically. The reason is because if they do well to health, if they cure colitis, if they do what, you know, if they have the effect we are buying them for, then at least in the United States, the FDA will take out that probiotic from the shelf and will make it a medicine, and you will need doctor's recipe, you know, doctor's permission. So that goes against the interest of the company. So the system has put us in this situation where the system favors to sell products that have not been tested enough. The only entity in society that could do those studies and publish them are therefore probably the outside the companies. Academia has the responsibility to show what is the effect of those probiotics that millions of people are consuming. I'm very skeptical because it simply we don't know the studies. If the companies have done them, they don't publish because their products will suffer in the markets. So it's a human system problem. But I think academia can help clarify which are the good probiotics and why is the differences are at the strain level. It's not even species. There seem to be strains that work and strains that do not work. But if we don't have complete freedom and money to do the research, if there are commercial interests, that really poses a problem.
Questions are coming continuously.
No, Amrita, now please wind up. Thank you.
Okay, ma'am. Thank you, Professor, for this amazing lecture, and thank you so much for giving such a valuable time. I would request Dr. Annalakshmi Chatterjee, ma'am, to express our sincere gratitude to Professor Maria.
1 minute, Maria. As you are the first scientist from outside India who have answered my invitation and accepted me. You may remember that 1 month ago I contacted you, and you just in one night you answered and said, when is your seminar? And thank you very much for accepting our invitation.
It's my big pleasure, and thank you all for your attendance.
Madam, on behalf of the organizing committee, I would like to thank you to be part of our webinar series. Your presentation was not only interesting, but also very effective, madam. Kindly accept our heartiest congratulations and sincere thanks for your outstanding presentation. Once again, I thank you for sharing your in-depth knowledge with us in this webinar series. Thank you, madam.
Thank you all. Thank you for inviting me, and thank you all for being there. Have a great time in the rest of your work, of your presentations.
Thank you, madam.
Your work on mother's milk, that attracted me.
I would now request Dr. Pubali Dhar madam to take over.
So now we are in the last part, that is concluding session. I have something to, I have things, people whom I should thank for this journey. I first want to thank my colleagues, my dear students, and all the registered international delegates from across the world on behalf of the Vice Chancellor, Madam of the University of Calcutta, and as the head of the Department of Home Science, University of Calcutta, I express my sincerest gratitude to all of you to convene on this virtual platform and become a part of a curative journey in nutritional science. Today, I'm proud to have with us Professor D.K. Bhattacharya, renowned oil technologist, ex-Ghosh Professor, Department of Chemical Technology, University of Calcutta, Adjunct Professor, IIEST Shibpur. Sir, you are my inspiration. He dreamt to work in the field of biological science, being a technologist. Being one of the students of Professor Bhattacharya, sir, I learned how subjects can be intermingled and researches can be done in different disciplines. This conference is my tribute to my teacher, who taught me the importance of interdisciplinary research. I am grateful to you, sir. Sir, you are my inspiration. In the concluding session of this highly productive international webinar series 2020 to observe the National Nutrition Month, I feel extremely honored and privileged to be able to conclude that this year we have successfully conducted an extremely engaging and thought-provoking lecture series that at some level also concerns women nutrition and augment awareness about areas that need to be addressed to improve the nutritional status of the general population. These 7 days, we have participation of several eminent and erudite academicians in nutritional and allied sciences from across the globe who gave us their very precious and valuable time to deliberate upon some of the very pressing research areas in the domain of nutrition. Ergo, it comes as no surprise that almost every known domain of life science research connects with the area of nutrition, and that all of us need to continuously revisit and upgrade our understanding of human nutrition. This webinar series, as was intentioned, I am certain must have opened us vistas of opportunities for young minds who wish to contribute towards nutritional development and research and become a part of a very exciting domain of health development. I, on behalf of the Department of Home Science, University of Calcutta, the entire organization, organizing members, Dr. Purba Chattopadhyay, Dr. Anna Lakshmi Chatterjee, my colleagues, and my other colleagues who supported me throughout this journey, students, together and on my own behalf, extend a very hearty vote of thanks to all speakers for gracing your important work and sharing with us your findings and opinions. We are very grateful and obliged to all the panel members and speakers of this international webinar series who responded positively to our appeal in order to educate us and inspire us with their outstanding achievements in their respective fields. In addition, these sessions have paved way for us with opportunities and guidance to improve the status of nutrition and health of Indian population through holistic measures. This international webinar cannot happen overnight and wouldn't have possible had it not been for the continuous and relentless efforts of my research scholars, students of the department, and teachers of food and nutrition in other colleges, the faculty members, and research scholars of different institutes who toiled so hard for several weeks before the commencement of this grand, ambitious event. The wheels start rolling weeks ago, and we have been fortunate enough to be backed by a team of very motivated, dedicated colleagues of our Department of Home Science who know their job and are result oriented. In addition, I would especially want to thank Niloy Chatterjee, being a microbiologist, worked as a computer expert, streaming the whole program. I want to thank Amrita Roy from CFTRI and Dr. Arun Dash from Indian Veterinary Research Institute, Kolkata, who have helped me all the time, whenever I was in a mess and I was in trouble. Dr. Tanmay Kumar De, postdoctoral research fellow, NICED, my former research scholars, Dr. Debojyoti Pal, assistant professor, Techno India University, and present research scholar, Orpita Banerjee, Srija Pal, Srina Dash, research scholars of Department of Home Science, Amrita Chakraborty, Debanjana Shane, research fellow, Department of Physiology, Madhusrita Dash, research scholar of the Chemical Technology, Department of University of Calcutta, Anadi Roy Chaudhary, my former scholar now at Srirampur College, to support us in making this event happen. I cannot thank all the speakers, organizing members, and participants enough who responded to our earnest appeal and encouraged us in making this event a success. We are in our debt, and we hope you will continuously support us, motivate us, inspire us in these educational initiatives of ours to shape a better world free from diseases and miseries. This pandemic has depressed us all, but a new hope of rejuvenation is still on the horizons, and together we can fight it. This webinar has been a burning testimony to this hope and conviction. A very safe and healthy year ahead to all. Now, thanks from Dr. Purba Chattopadhyay. She will end and she is with me, and Dr. Anna Lakshmi is with me from the from uh, from the throughout this journey. So I hand over the mic to Dr. Purba Chattopadhyay.
Thank you so much. Am I audible? Am I audible?
Yes, you are audible and visible.
Okay. We had been discussing food in the this 7 long days of food, which is a biological necessity as well as biochemical in composition. It is a cultural identity. It is a comfort zone. It is a emotion. Food which at times has caused war. Food which causes hierarchy in our society. This food, which has so many diverse aspects, is under one particular string united. That is the string of nutrition. It provides the nutrition to one and all across the culture, across our diversities. It provides us with the very basic of our survival. This nutrition or portion or pushti has been celebrated through the month of September in India, a country which still has lots of malnourished children. The dual focus of identification and tracking of children with severe acute malnutrition and Poshan Ke Liye Paudhe were this year's particular themes. These were through this nutrition week or celebration or nutrition month, Poshan Ma's celebration in our department. Did we exclusively focus on these two themes only? No, we were actually trailing through many more diverse kind of topics. The thing is that let's just think of a string instrument like a sitar or say a guitar. The strings, each of the strings has a particular note, and within the strings there are so many notes or so many scales of a particular note. To a novice, when he or she touches the instrument, it produces noise. However, it is the maestros who, when they handle the instrument, they produce beautiful music. I thank all our eminent speakers across the globe, across our length and breadth of our country, from the eminent institutions, including our own university. I thank them because they were the maestros where they have brought in the diverse field from where we could relate it ultimately to the science of nutrition. We feel enriched. Our heartfelt thanks to all of you. Next, I would like to thank the very basic organizers without whose support this particular event would not have been possible. Our Vice Chancellor, our Pro Vice Chancellor Academic, our Pro Vice Chancellor Finance, and our Registrar. They had been the backbone of our support. We thank you all from the core of our hearts. Next, we come on to the teachers who are our support system and the non-teaching staff. We all thank you so much. And the students and all the participants who have been again a source of inspiration for organizing an event in the scale so large. Lastly, a particular special thanks to Shrija Pal, Niloy Chatterjee, Dr. Tanmoy De, Urpita Banerjee, Amrita Chakraborty, Debanjana Sen. You are students and you are scholars. We can wish you all the best in whatever you do. You are very hardworking and we are so proud of you. Next, at last, this is the particular moment I was waiting to share with the audience. That is, whenever there is a successful event, there is one particular dream behind it, and a person who had actually dared to dream it that large. That is our own head of the department, Pubalidi. Thank you for imagining a thing in the scale of this big canvas. We feel enriched. I am so proud. I am so happy. And actually, words are failing me that you are my colleague and you are always like an elder sibling to me. Thank you all so much for this grand event. Hopefully, we will again meet. Hopefully, Pubalidi will again dream of such a great event, and again we will be under the auspicious of one particular roof. Thank you all so much.